Hello, everyone. On behalf of the organizing committee, I would like to welcome you all to the third day of this workshop, First Human Cell Atlas Latin America Single Cell RNA-seq Data Analysis workshop. So we are on our third day. For us who are joining us for the first time, this is the second event held in collaboration with the Human Cell Atlas targeting the Latin America region. However, it's open to the whole scientific community. So we welcome our colleagues from Europe, from the US, and from different parts of Asia. So the idea of this workshop came up last year during the first event of the Latin America Human Cell Atlas. And we have organized this event with colleagues from Mexico, Chile, and Brazil. So both the organizing committee and the scientific committee include people and researchers from these three countries. It is our intention to have a follow-up, including colleagues from other countries from Latin America. On the second week, we have a hands-on workshop, and trainers are located in Brazil, Chile, and Mexico for this part of the workshop. We would like to thank the Human Cell Atlas crew, people who were together with us since the beginning of this, the organization of this event, especially Christine, Luke, Tracy, Samantha, and John, the institutions that have supported us financially and with infrastructure. So our program for today looks like this. Please be aware of the time zone where you are from so that you're able to join at the proper times. We have 2 sessions in the morning, a 45-minute break, 2 more sessions, and a half-an-hour break, followed by project design discussion. This part is for people who are going to be joining us and have been selected for the second week of this event. For this part, you will be getting, as usual, a specific link, not the same link from this meeting in the morning and afternoon. I'll be hosting you now for the first sessions, and Enrique Hernandez will be hosting our afternoon sessions. So welcome you all. With this, I would like to introduce our first speaker. So Gerda, she's a PhD student in Sam Bejati's group at the Sanger Institute. Her research focuses on the relationship between fetal development and various cancers, including but not limited to neuroblastoma. Thank you so much, Gerda, for joining us here today.
Thank you very much for having me. I will just share my screen. One second, sorry. Are you all able to see my screen?
Yes.
Yep. Yes. Okay, fantastic. Thanks a lot. So, hello everyone, and welcome to this talk, which is going to be about dimensionality reduction and clustering in single-cell RNA-seq. Before I start on the subjects at hand, I want to tell you a little bit about me. This is a picture of me looking all bright-faced and excited in the first year of my PhD. Obviously look a little bit different now, a little bit more tired. I am a PhD student, as you already know. I'm currently in the final year of my PhD. My thesis is now upon me. And it just so happens that I actually started my PhD in something completely unrelated to single-cell RNA-seq. But my supervisor left a year into my PhD, so I completely switched subjects and have been working on single-cell for sort of a year and a half now. And the reason that I'm telling you this is that if you are a beginner, like I was a year and a half ago, you can definitely get into doing the sort of analysis analysis, even without a maths background, which I don't have personally. And even if you don't have an in-depth understanding of all of the methods, because they are generally quite sort of maths heavy, but you can, it is possible to do it. And if you're not a beginner, then perhaps you can gloat that you know more than me by the end of this talk. In the session, you will learn about PCA in a little bit more detail. I think it's really important to have at least a general level of understanding about PCA, given that single-cell RNA-seq essentially doesn't happen without a PCA. I will also go through UMAP and TISNE, although I will focus on UMAP a little bit more. And then I will also talk about various clustering methods, and also a short section about cluster annotation. And through all this, I will also try to cover some of the best practices during sort of these steps of single-cell RNA-seq analysis. So why do we actually need to use dimensionality reduction? So in single cell, data is highly dimensional. So we have lots of genes and lots of cells. And it is actually really hard to visualize anything well in more than 3 dimensions. And I've kind of just shown this plot here to illustrate that. So we have an X, a Y, and a Z axis here. And then we have a 4th dimension that is represented in color. And it's honestly just really hard to understand what is going on. And at various points, the points are hidden behind other points. And it's sort of really hard to make sense of something like that. So we need ways of looking at data that actually make sense for the human brain. In addition to that, the amount of data is huge. So when first single cell data set was generated, I think in 2009, it had only a handful of cells. And now I think probably a year ago, 10x generated a data set that was 1.5 million cells or something like that. So there's a huge amount of data. We need to think about computational power that is available to us. And actually, I just wanted to share with you, I took a screen grab of it this morning. This is our cloud computing server. And someone was clearly either having a sleepless night running a job that required 140 gigs of RAM, or I mean, hopefully it wasn't a sleepless night and they were just running an overnight job. But the point is that it's obviously the amount of memory and processing power that you need is massive. So we basically need to have methods that reduce the computational resources required. So in the first part of this talk, we'll essentially talk about how we go from this, which is a count table. So we have genes as rows and cells as columns, which can be massive. So in this case, in this example, it's a pretty small matrix. I mean, the full matrix is sort of the size of 20,000 genes and 10,000 cells. And what we want to do is we want to go from this to a 2-dimensional representation of that data, which has tens of thousands of points. So the first step in that, in getting there, is principal component analysis. So principal component analysis is a technique that is used to emphasize variation as well as similarity and to bring out strong patterns in a data set. And sort of for us, the more important thing is it reduces the number of dimensions. So essentially the amount of data that we need to work with. So let's have a look at this toy data set. I've included this link where part of the tutorial is taken from. So feel free to screen grab or just write it down and have a look in your own free time if you do have the time. But back to this example at hand. So let's say we have a very simple data set. We have 2 cells and we have 4 genes. And we can represent one of the samples on the x-axis and the other sample on the y-axis. And we just kind of plot the gene expression for those 2 samples for these 4 genes. So you could draw a line through the data in a direction representing the most variation, which is on the diagonal in this example. And the maximum variation in the data set is between the genes that make up the 2 endpoints of this line, so genes B and C. And we also see the genes vary somewhat above and below that line. So genes A and D, and we can draw another line through the data that represents that second most amount of variation, since this plot is in 2D. And so the genes near the ends of each line would be those with the highest variation. So these genes have the greatest influence on the direction of the lines mathematically. And if we had 3 samples or cells, then we would have an extra direction, and then we would have a variation in 3 dimensions. And if we had n samples or n cells, then we would have an n-dimensional representation. So, and that is essentially what principal components are. So you can think of these lines as the axes that represent the variation. These axes are essentially principal components, with PC1 representing the most amount of variation in this data and PC2 representing the second most. And once these PCs have been determined for a data set, we have to figure out how each cell fits back into that context so that we can visualize the similarities or differences in an intuitive manner. So scores are calculated essentially for all sample PC pairs, and I'll describe how you actually do that. So the first thing that happens is each gene is assigned an influence score based on how much it influences each PC. And genes that did not have any influence on a given PC get scores near 0. So something like gene B doesn't have a lot of influence on PC1 and gets a small score. While genes with more influence receive larger scores. So these are the genes that sort of are at the ends of a line. So in our PC1, that's gene B and gene C. So they will have the opposite. So they'll have high values, but they'll be the opposite values. So gene B has a score of -10, and gene C has a score of +8. So once we do that, we actually sort of we kind of calculate the total effect. So you kind of multiply the gene value by the sort of the influence on the principal component, and then you sort of have a PC sample pair and one score for each pair. So you just have one value for each sample, and then you can represent that in a 2-dimensional plot. So when you have a real single-cell RNA-seq data set, you obviously first, so the standard matrix will be, maybe 20,000 genes. And the first thing that we do is we do actually reduce the dimensions a bit first by selecting the most variable genes. This is a standard step in single-cell RNA-seq analysis, basically selecting the genes in your table that explain sort of the most of the variation. And in this sort of our imaginary toy data set, let's say we have 10,000 cells. So in accordance to our previous toy example, we have a 10K dimensional plot of our 2K most variable genes. So then to create PC1, a line is anchored at the center of this 10K dimensional cloud of dots and is rotated in 10,000 directions, all the while acting as a mirror on which those original 2,000 points are projected. And this rotation continues until the total distance among projected points is at the maximum. And that rotating line now describes the most variation amongst those 2,000 genes and is fit to be PC1. And then the second most amount of variation is fit to be PC2, and so on and so forth. This sort of 10K dimensional rotation is very hard for human brains, but thankfully very easy for computers. However, just think back to the toy example we've been through already. Essentially, what you're doing is you're summarizing the effect that genes have on variation between cells into a single value for each cell at the point where the overall level of variation is at the highest. So let's say you calculate your PCs. You can calculate essentially as as many as you want. But the question is how many of them actually represent the variability in your data, and therefore how many to use for downstream analysis. And if you've ever been through a Seurat tutorial, which many of you probably have, you will have been offered something like jackstraw or elbow plot to determine how many PCs to use downstream in your analysis. And I will just quickly go over these methods. So in jackstraw, what it does is it kind of randomly permutes a subset of your data, and it calculates projected PCA scores for these random genes. And then it compares those PCA scores to the random genes with the observed PCA scores to determine statistical significance. And the end result of that is a p-value for each gene's association with each principal component. So we essentially identify significant PCs as those that have a strong enrichment of low p-value genes. However, you still have to decide how many PCs are actually significant. And this is hard when the dimensionality of your data set is high. And in the elbow plot, essentially all it is you're plotting the amount of variation that each principal component is explaining. And as we already said, principal component one will explain the most variation, and then principal component two, the second most, and so on and so forth. And you essentially just plot how much variation in your data the principal component is explaining. So in this example, so as I say, these both are taken straight out of Seurat PBMC tutorial. So in this example, we can kind of see that you have this elbow. So the level of variation that each PC is explaining kind of tapers off. So it would suggest that the true dimensionality of the data set is around 9 or 10 PCs. So as I say, these are very nice examples from PBMC where it's really easy, but I don't know about you personally. I've had plenty of data sets that I've worked on where, you know, you calculate PC scores with jackstraw for like 40 PCs, and the 40th PC is somewhere there, and you're like, how many PCs do I use? And in addition to that, jackstraw also take ages to run. So 40 PCs takes something like 40 minutes or something like that. So and even then, like, even if you can calculate a lot of them, it's not always helpful. So and it kind of feels like you're almost arbitrarily picking your principal components. To be honest, I would say that in the last sort of, I don't know, 10 or 20 single cell RNA-seq papers that I've read, no one even justifies the number of principal components that they're using in downstream analysis. They'll just say we used 50 PCs, and that's that. Doesn't seem to be a requirement to actually explain your reasoning. But there is actually an alternative option, and it's called MCV, or molecular cross-validation. So briefly, what this method does is it randomly apportions the counts from cells in the dataset into two groups, each sort of simulating a shallower but statistically independent measurement of the original cell. And the accuracy of principal component then can be evaluated by fitting it on one of the groups, so training on one of the groups, and then comparing the PCA output to the other group. And the nicest thing about this method, probably the most useful, is that it gives an average error. So it's basically a metric that says how accurate the sort of the training and the validation data sets match, and it has an absolute minimum value. So essentially, as you run it with a different number of PCs, your error will at some point increase to the point of minimum. But as you increase the number of PCs, it can actually get higher. So it actually just outputs one single value, which is like, that is the dimensionality of your data set. Of course, it has limitations, just like any other method. But if it's something that you're interested in, if you, like me, have been dissatisfied with running jackstraw and it's still not really making sense, and you're not really getting any PCs that are not enriched in statistically significant genes, then I suggest it's something that you try out. You can look up MCVR, which is an implementation for MCV in R that is actually written by my colleague in Sam's team. So let's say you did all of this, you determined the dimensionality of your data set. Unless a data set is very homogeneous, which it definitely can be if you're only looking at one particular cell type, if you're sorting or selecting for just one cell type, or you just have a fairly homogeneous tissue, it could be that 3 PCs are enough, but that's generally quite unlikely. As I've mentioned before, most of the data that I've sort of worked with has been the sort of the correct number of PCs to use has been around 50. So we kind of run into the same problem that we ran in the beginning, where we can't really look at things in more than 3 dimensions. I mean, it's still very useful to plot your cells along the PC score, so PC1 against PC2, because there are some things that can show up, like it's possible that if you plot your cells and then you color by batch, for example, you can see a very clear separation among PC1, and that actually tells you that your batch explains the most of the variation in your data set. Or the largest sort of proportion of it. So it's still definitely useful to look at PCs, but in the end, we want to look at our data in 2 dimensions. And this is where UMAP, which stands for Uniform Manifold Approximation and Projection, an absolute mouthful, and TSNE, T-stochastic neighbor embedding, also an absolute mouthful, come in. So I don't really want to get into the maths of these methods, primarily because I don't really have enough maths knowledge to understand them either. But in my layman level of knowledge, what these methods essentially do is they construct a high dimensional graph representation of the data. And then they optimize a low dimensional graph to be as structurally similar as possible. And whenever you're running, for example, the run UMAP function in Seurat, I mostly work in R, so that's why I have my little R function here, or whatever the equivalent is in Python. You have to specify 3 main parameters when you do run UMAP. The first is the number of dimensions, then the minimum distance, and the number of neighbors. So yeah, so this is my quick and dirty R processing function. When I get a data set I don't know anything about, I run it through this, and then I have a look at it. So the number of dimensions is pretty self-explanatory. You've already sort of decided what the dimensionality of your data set is, and then you kind of just run with that through the rest of your analysis. And the reason that we do that, you could in theory run UMAP on your whole matrix, but it will probably take forever. So we first reduce the number of dimensions to the point where we have PCA, and then we run UMAP. So one thing is it takes a long time, and the other thing is single cell data is inherently noisy, and PCA helps reduce that noise a little bit. So PCA, fine. Now the minimum distance and number of neighbors parameters are key. And so instead of kind of droning on about this, I think it would be much cooler if I can show you what you actually need to know about these methods. So there's this really cool site. Again, have either, I don't know, take a photo or a screen grab, or just write it down. But I encourage everyone to sort of go there and to play around with in their own time, because it actually just shows what UMAP and TSNE can do. And I will mostly focus on UMAP because I think it's sort of more relevant, but I will touch on TSNE as well. So there we go. Back to number of neighbors and minimum distance. So the most important parameter in UMAP is the number of neighbors. And that number is the number of approximate nearest neighbors used to construct that initial high dimensional graph. So effectively, it controls how UMAP balances local versus global structure. So if you have low values, they will push the UMAP to focus more on the local structure by constraining the number of neighboring points considered when analyzing the data in high dimensions. And high values will push towards representing the big picture structure while losing fine detail. So I'm going to show you this really cool 3D mammoth data set and its UMAP projection. So this is our mammoth. It's in 3 dimensions. So each point has an X, Y, and Z value. So this is the sort of the original data set. And then we represent it in 2 dimensions using the UMAP algorithm. So because we want to just have a look at number of neighbors, I'll set the minimum distance to somewhere in the middle, and then we'll vary the parameter. So as you can see, when the number of neighbors is really small, so 3, you basically don't have any sort of global structure. It's all local. And when you actually look, so it's really cool when you point at points here, it also points them out here. So you can kind of see that they're all really, really close. So it's definitely conserving the local structure, but it's not conserving the global structure as well. Although you can actually, it's really cool. You can see that all of the yellow dots are actually close together. So it's, in some sense, that global structure is still represented a little bit. So as you increase the number of neighbors, little by little, you start seeing more of that global structure. So you can kind of see the representation, the way that it kind of looks a little bit on the actual mammoth in 3D. And as you increase the number of neighbors, you get that more and more, to the point where when you get it quite high, it actually looks kind of like a mammoth, if you kind of were looking at it from above, right? So yeah, so I mean, you still have relatively decent local structure representation. Like as I move the sort of the cursor along the tusk, you can see that it is moving along the tusk in the 3D plot as well. But you also have some sort of, you know, differences where you have like this leg, which is a little bit here and then a little bit here. And so the sort of the local structure essentially just isn't as well represented. So that's number of neighbors. It's really important whenever you run UMAP. This is the kind of thing that you have to remember. So basically, low number of neighbors, local structure; large number of neighbors, more global structure. And then the other parameter to talk about is minimum distance, or the minimum distance between points in low dimensional space. So this parameter controls how tightly UMAP clumps points together, with low values leading to more tightly packed embeddings, and then larger values leading to sort of less tightly packed embeddings. So the larger values of minimum distance will make UMAP pack points together more loosely, focusing instead on preservation of the broad topological structure. So in this case, I'll sort of set the number of neighbors to, I don't know, 50, and then I'll vary the minimum distance so we can have a look. So yeah, low minimum distance packs points really, really closely together. And then as you increase it, it's more and more loose. It still has some of that representation of the high level structure, but it's obviously a very different look. So essentially, running the UMAP is kind of, you have to have a balance between these two parameters. And in I think like the standard in Seurat and probably in other software packages is number of neighbors of 50 and minimum distance of 0.5. So, you know, it's pretty good. As in it looks quite a lot like a mammoth from above. It's not a bad representation. However, you need to remember that this is 3D. If we are doing this with a data set where we've calculated 50 PCs and we've decided that 50 PCs is the dimensionality of our data set, then you can imagine how different that is. but it's kind of the best we have. So that's kind of the two parameters for the UMAP. And then the other thing that I want to briefly touch upon is UMAP versus t-SNE. So the main difference between UMAP and t-SNE is this ability to preserve the local versus global structure. And what I want to show you is, so in t-SNE, you only have this one parameter called perplexity. And that is it kind of does the job of both number of neighbors and minimum distance. And if you have a look at here, you have these yellow clusters, and all of these dots, all of the yellow dots that are, so in the yellow clusters that are scattered throughout this, they're all very closely together, even when you look at the at the UMAP, which does conserve more of a global structure, but they're all scattered. Whereas previously, when we looked at this, even if you had a really small number of neighbors, all of the yellow points are all together. So you still have a little bit of that global structure, whereas in t-SNE, it just completely falls apart. And in t-SNE, as you sort of increase that perplexity parameter, the running time increases massively. By the time you get to the point where you see something that looks like a mammoth, it takes at least an hour to run. And again, I'm going to remind you, this is 3 dimensions. When we're doing single cell, we have like 50. So yeah, I mean, when we look at the UMAP, doing something like, as I said, pretty normal for single cell, number of neighbors 50, minimum distance 0.5, takes 2 minutes, and that is a pretty cool, pretty good global representation of the data set. So right, let's continue. So we've done our UMAP, we've constructed our 2-dimensional representation of the data, but how do we actually identify which cells are an actual grouping? If we look at this unlabeled UMAP, we can see that there are clouds of cells that are similar to each other. But as I've alluded previously, UMAP structure isn't always perfect, and oh, my apologies. And the balancing of local versus global structures is tricky, especially as I say, we don't have 3 dimensions, we have 50. So, so to go from this, which is our unlabeled UMAP, to this, which is our clustered UMAP, we need to run a clustering analysis. And the goal of clustering is essentially to discover natural groupings of a set of objects. And in single cell RNA-seq, we define those groups on the basis of the transcriptome. There are 2 main types of clustering. There's supervised clustering, which we kind of don't care about as much, because in supervised clustering, you kind of have prior knowledge of what you're looking for and which cells are likely to have which labels. Whereas in unsupervised clustering, we don't have that prior knowledge on the number of clusters, and we need to identify groups based on similarities without actually knowing the labels. So some of the most common clustering methods in single cell RNA-seq are graph-based, K-means, and hierarchical clustering. And I will just go through these with you. And we'll kind of talk about which packages they're actually used in. So the first thing that we'll talk about is a graph-based method. Sorry. Very dry throat, talking a lot. It's been a while. So in essentially in graph-based methods, we construct a graph using Euclidean distance. The PCA thing, like that's just the same thing that we've been talking about before. We're not constructing a graph from the whole expression matrix. We first reduce the number of dimensions. That's all it means. So yeah, so we construct a k nearest neighbors graph using Euclidean distance. Euclidean distance is just the straight line distance between 2 points, nothing scary. And then another kind of distance metric used, Jacquard distance, is used to refine edge weights. So essentially saying how important this distance is versus that distance. And then a Levein algorithm is used to essentially iteratively group cells together. So the algorithm finds a group of cells that it labels as sort of as a group, and then once that happens, it goes through a second round to the point where it stops. And essentially, if you are running find clusters function in Seurat, I know a little bit more about R-based methods because that's mostly what I work in. But you basically have to specify a resolution parameter, which essentially affects at which point the algorithm stops in the sense of how fine the clusters are. So if you have high values of a resolution, then you'll have more clusters that are probably smaller, so more granular. And when you have small values, then you have sort of larger clusters. So that's essentially, this method is what is used by Seurat and scanpy. So probably the two most popular ways to software packages to analyze single cell data. Then the next clustering method is K-means. So this is something that is used in SC3. And so again, same as with graph-based methods, you don't actually kind of work in the actual count matrix. You work in a PCA space, and we don't really have to worry about that. The sort of the whole thing about SC3. So in K-means, the way that it works is that you have to specify K, which is the number of centroids, or in other words, starting points for the algorithm. And there are methods to estimate the best K, but it's not exactly an exact science. But in SC3, the way that it works is that the algorithm actually uses a range of Ks. So it will run the algorithm with K equals 2, K equals 3, K equals 4, and so on. It's your choice. And then it comes up with a consensus clustering. So it essentially, so it switches up the K, and then it checks whether the cell stayed in the same cluster or whether it went into a different one when different K is picked. And so that information is kind of all taken into account to then assign cells to cluster identities. So the final method of clustering that I will talk about is hierarchical clustering, which is the simplest one. And with hierarchical clustering, cells are essentially grouped together based on whether the transcriptome is similar. So in agglomerative clustering, cells are iteratively grouped together until they belong to one cluster, and the results can then be interpreted as a dendrogram. So for example, in this case, we have A, B, C, D, E, and F, whether they're cells or whatever else. And the algorithm sort of pairwise compares all of them and says, okay, D and E are the most similar and B and C are the most similar. Then it compares B and C to anything that's not been grouped yet. And so it goes and until it kind of reaches a consensus. So so hierarchical clustering doesn't really give a number of clusters as such. You can just cut the dendrogram at each level and sort of decide where your clustering is. So it's not, it doesn't actually give you a very precise cluster assignment. So there's a really good paper by Vlad Kiselov, who is at Sanger, and it summarizes various clustering algorithms, including their strengths and weaknesses, and I encourage everyone to have a look at it. It's really important to know your data. If you have a large data set, you probably don't want to go with hierarchical clustering or SC3 because they're computationally heavy. So actually, Vlad wrote SC3, and he very humbly puts out its limitations in his own review paper. So yeah, so it's not very scalable for when you have large data. Something like hierarchical clustering, it actually, the amount of power, computational power needed increases quadratically with the size of the data. So that is also not very good for where you have really large data sets. Whereas something like, PCA plus graph-based methods like scanpy, Seurat, phenograph, they're fantastic for very large data sets because they're very scalable to large data sets, but they may not be as accurate for small data sets or if you have really rare cell types. So it's really important to know your data. If you have something really small, if, I don't know, you're studying development, I don't know, from 1 to 16 cell embryos, and you don't actually have that many cells, maybe it makes sense to look at hierarchical clustering or SC3. And maybe then it doesn't make sense to look at Seurat or scanpy. But if you have a million cells, then Seurat or scanpy is probably your best friend. So yeah, it's just important to have a look at your data, kind of know what you're dealing with, have a look at review papers like this to know what to do. So now I want to switch gears a bit, although this isn't strictly about clustering. The analysis doesn't really end when you have your set of clusters, and cluster annotation is a really important step, and they kind of go hand in hand. So the thing that I'm showing here, this is actually my own data set. Well, I didn't produce it, I just analyzed it. But this is fetal adrenal gland, single cell from a bunch of different embryos. And this is the way that the data looked like before QC, and this is what it looked like after. So obviously quite a stark difference. And the reason that clustering kind of ties into all of this is that generally your clusters are going to be cells that are similar, pretty obvious. But a cluster doesn't necessarily equal a cell type. And sometimes clusters are actually a group of cells that are completely different cell types. They're not related, but they're similarly bad quality. So for example, doublets or cells with really high mitochondrial content, et cetera, et cetera. So hopefully by the time that you got to the point of doing dimensionality reduction and clustering, you will have done your QC, but there are some things that you can't really see until you look at it in your final UMAP. So something like doublets will generally show up in separate clusters. There are sometimes clusters that are specifically, as I said, to do with QC metrics. Even if you have, for example, decided to set a particular mitochondrial gene threshold and you cut it off, but then you have a look at your UMAP and you find one cluster where consistently all of your cells are on the higher end of the mitochondrial content, then maybe that's something to sort of have a look at. Sort of looking at your data after clustering, as I say, can actually be really informative from a QC standpoint, and not just for labeling cell types, but for identifying problems in your data set. And if you actually look at the previous UMAP and compare it to this one, so if we have a look at this and compare it to this, you'll notice that actually a bunch of clusters are grouped in categories. So, mesenchyme is actually clusters 11, 18, and 23. And so on and so forth. And the reason that we've done that is they actually encompass, so for some of them, they actually encompass a bunch of different cell types. So leukocytes are obviously, B cells, T cells, whatever. But for some of them, like actually the mesenchyme, they actually represent different gestational ages, for example. So sometimes, as I say, your cluster doesn't necessarily represent a different cell type. Same for adrenal medulla. This is the bit that we were actually interested in for the kind of analysis that we were doing for our paper. And there are actually two clusters clusters here, so cluster 19 and 20, but there really wasn't anything in like, you know, in these cells versus these cells that would have warranted separating them as different cell types. And you often see that in single cell papers. They have 5 clusters of essentially the same cell type, but they will just say, oh, this is hepatocyte cluster 1, hepatocyte cluster 2, without ever actually explaining what they mean, or whether it's just a little bit of a batch effect or noise in the data, which is fine. There's nothing wrong with labeling your clusters as the same cell type if you really truly believe that they are the same cell type. And so let's say you've come to the point where you know, you've used your UMAP to look at certain QC metrics. You're kind of happy with all of the downstream and all the upstream analysis that you've done. So how do you actually annotate clusters? So the 2 main broad ways are marker-based, which is where you use markers from the literature or cluster defining genes using certain methods like find markers in Seurat to annotate your data. And the other way is automated, which is where you use a previously existing data set to predict the labels for your data. So marker-based annotation is obviously pretty simple. You just find some markers. For our paper, we just found this paper from 2006 where they did laser capture microdissection and then RNA-seq, really state of the art in 2006. And they came up with a bunch of markers for, we were specifically interested in these 2 clusters. So I'm going to spoil it for you. These are chromaffin cells and these are sympathoblasts, but we kind of didn't know it back then. So we were looking at sort of both human and mouse markers. And actually there was, at the time, there was no single cell data for human from the fetal adrenal gland. So we ended up looking to mouse quite a lot. And this is kind of where the automated annotation comes in. So essentially, the way that automated annotation works is that you have a data set. It could be mouse, could be human, but something that is actually relevant to whatever single cell stuff that you're doing. And you train the model, a predictive model on that data set, and then you try to predict the labels of your own data set based on that data set. So it's actually something that we did for our paper. We used a mouse data set as a reference, and we predicted the labels to our own adrenal medulla. And it was essentially exactly what we expected. So mouse SCPs matched to human SCPs, mouse bridge matched to human bridge, mouse sympathoblasts matched to human sympathoblasts, and so on. This was actually quite a controversial thing because there's this one marker in mouse called CAR-TPT that is expressed in I think it's in chromaffin cells, but in humans it's expressed sort of in the opposite cell type, and it kind of, despite that marker, when you look at the overall transcriptome using automated annotation, it kind of it still matches that specific cell type. So yeah, so something like canonical annotation from Seurat, so transfer label function, I think is the one that does it, or something like logistic regression, which is what we did. If you want to read a little bit more about logistic regression, you can find it in my paper from this year, or Matt Young's paper from 2018. So it's kind of the same principle. You kind of train a model, and then you predict it. You predict the cell labels for your query data set against the training data set. So I think I'm going a little bit over time, so I'm just going to summarize real quick. So dimensionality reduction, you pick some variable genes, you do the PCA, then you do the UMAP, you have your two-dimensional representation. Finding the dimensionality of your data set is tricky, but there are some tools available to do that. The most important thing about clustering is picking the right algorithm that's essentially right for your data. When you annotate, cluster is not equal cell type. Don't assume that. Make sure that you try a few different methods. And there are various tools for cluster annotation, and I would recommend using sort of a combo of looking at markers, but also looking at, if you have the opportunity of looking at automated annotation as well. So thank you very much for listening. And if you have any questions, I will take them now.
Thank you so much, Gerda, for such a great talk. That was really, really good. It's amazing how from the beginning you told us about your background and how much you've learned during this period, right? And how much you are aware of all that goes on in the background of what you're running. It's really great talk and inspiration, I hope, for all our students who are here in this meeting. We do have quite a few questions from the audience, so I'm going to start with the most voted ones. So first one, hello, great presentation. I have a question. How do you manage to leverage variability with clustering when you don't know a priori how many clusters will be representative of your cell populations? You kind of talked a little bit about that, right? But
Yeah.
Is there any kind of heuristics that you have found useful or most useful?
Honestly, no. And this is like, I think it's actually this big thing that a lot of people will have struggled with RNA-seq, because you have a data set and you run your thing, maybe you run it with the standard parameters, maybe you try to play around, but in the end, you're not actually sure whether each cluster is a cell population or not. And I've actually talked to a lot of people who have done single cell RNA-seq, some people that are the same as me and maybe don't have the sort of the right background, so to speak. And it's basically always tricky. You just have to know your data in and out. You have to look at the cluster markers. And essentially, in the end, it's basically deciding like, okay, even if there's a difference in expression of these two markers, I don't think they're the same cell type, or I think they are the same cell type. So yeah, I'm sorry. I think it's quite a hard thing to do. I think actually potentially automated annotation can help with that, because if you manage to find a different single cell data set that actually has some labeled cell types, and especially if you know that it's a really good quality data set, then that can be sort of helpful, because then it'll just predict the labels, and you can kind of have that as at least some kind of basis for it. So yeah, sorry. No heuristic that I found useful.
I guess there's a lot of biology involved in that, so maybe it's always going to be hard to have.
Yeah, for sure.
So next question. Great presentation. That's from Samantha Kuwada Teixeira. Great presentation. Thank you. That's from Samantha. Okay. Regarding wait, somebody's changing. Okay. Regarding UMAP method, do you suggest to start with default values and analyze the result, and after come back and redo the dimensionality reduction until your data present results with biological meaning?
Yeah, so I would say that the good thing to do is you can actually do that sort of pretty simply when you run it. Try a few different parameters at the same time. So basically do a combo of different number of neighbors and different minimum distance. And honestly, it's kind of difficult to just be like, to say, oh yeah, and just pick whatever looks right. But that's kind of how it works. There's not really a very good way of deciding for, what is kind of representative and what actually fits. I mean, as actually with clustering, the thing to do is then to see if it biologically makes any sense, and if it really doesn't, then maybe something's not right. So yeah, I would say definitely try both, kind of a combination of running different parameters, maybe at the same time plotting, whatever like other covariates you have, batch, I don't know, like predicted cell types, whatever. and then actually just have a look at the whole picture altogether.
Okay, great. From Tiago Lubiana Alves, excellent talk. Louvain does not provide a cutoff for choosing an exact number of clusters like hierarchical clustering does. I have a case in where I want to divide my cells in exactly 2 clusters. What you would do in a case like that?
Well, so I guess that depends. Like, why do you want to divide your cells into 2 clusters? If it's because you know that they're like, 2 separate experiments, or because they look separate on the UMAP or something, then just do it. You don't necessarily have to use an algorithm to tell you what you already know if it's something that you already know. Like, yeah, Louvain is not going to provide you like it, because that's what it is. It's an unsupervised algorithm. You just kind of select some values for sort of, I guess, the level to which it's going to run, but it's not exactly it's not, as you say, it's not a hierarchical clustering. So yeah, I'm not entirely sure what you want to do here, but you can just divide your cells into 2 clusters. Like, if you've done an experiment and you have, like, I don't know, one condition and another condition, and you want to compare the cells between those 2 conditions, that's fine, nothing wrong with that.
That's perfect. So from Juan Sebastian Arcila Barrera, thanks for the presentation. There are plenty of machine learning methods supervised for annotation of data. Is there any preferred method for single-cell RNA-seq data annotation? Use logistics regression in your paper. Other methods like supervised vectors machine or neural networks would work well too.
Yeah, so I don't think that there's any preferred method. I think that well, actually, there is a preferred method. If you look at most of the single cell papers that come out, people look at markers. Literally, people trawl through the literature, they find the markers, and then they annotate the data. And there's nothing wrong with that in general, except sometimes there is. So I didn't talk about it too much because it wasn't the scope of my talk, but in my own data, we had this problem where, as I've touched upon it briefly, where we had one marker that was sort of swapped around for the expected cell type in the mouse data in comparison to human. And actually, some people have made mistakes and got the annotation wrong in their paper. And yeah, so you have to, I think, especially now, we have methods that can actually take the whole transcriptome into the account. So you should also use them. So yeah, something like logistic regression or correlation, a correlation analysis. I mean, I'm not hugely familiar with any of the sort of like, I mean, higher level isn't the right word, but something like neural networks is just a little bit too black box for me. So I'm sure they would work fine. I just don't know. I just don't know if any. But yeah, I mean, if you find something that works, shoot me an email. Let me know.
Great. Another one here, great talk. When using data integration methods, a new reduction is usually generated, harmony and MNN, for example. Can these new dimensions be considered equal to PCA?
Yeah, so yeah, so essentially what you do when you like when you're doing like batch correction with harmony, you do that on essentially those new dimensions. So I have to confess, I'm not particularly knowledgeable on batch reduction because I hate it. Batch correction, that is, not batch reduction. I think that a lot of the time when you do batch correction, you don't necessarily have to. Like, I know it doesn't look as nice when all of your clusters are not necessarily in the same place. I mean, you should probably do it if you have, like, I don't know, two patients, and like, and B cells from one patient are closer to, I don't know, epithelial cells from that same patient than they are to B cells from the other patient. But if you have 2 B cell clusters that are close to each other, but they're not the same cluster, that's okay. So, and I think that's people generally, I think, overcorrect when it comes to batch correction. But yeah, those dimensions, they're essentially like, that's what those new reductions are. They're just like a PCA score that is like slightly more integrated. I mean, I don't know the maths behind it, but yeah, they're essentially equivalent to the PCA scores.
Great. We did have some talks yesterday that addressed the overcorrection of batch effects too.
Yeah. So I'm sure that, yeah, definitely not an expert in batch correction. Sorry.
Okay, next one. Great talk, and thanks for the encouragement. I am also a beginner with no math background. Could you please suggest some resources or guide on how to move about? Also, how do we get to know that now we are sure the results are correct and we are not going in wrong direction?
Yeah,
that's kind of tough.
Yeah, it's tough. So regarding the resources, let me think. So some of the things that I've kind of shown, so there's like that tutorial that I based my talk on for PCA, like that's really good for something like PCA. Actually, YouTube is not bad for these like simple mathematical concepts. There's, people like Khan Academy and stuff like that that actually will explain it. Go to courses if you can, if you're able, and in your institute or institution can send you. There are generally courses. I actually ran one pretty recently on single cell analysis, where we basically tried to be as inclusive as possible of new starters. I mean, we kind of require that people have some level of R, but in terms of the actual concepts. So yeah, I mean, online is good if you can find someone who can mentor you, even if it's maybe there's no one in your team or or even like sort of in the nearby community. If you can try to even find a researcher from maybe a different team or a different institute that could maybe help you. And yeah, this whole thing about not going into wrong direction, it's tricky. It's very, very tricky. But yeah, I think for that, the best thing to do is, if you can, try and get someone on board that has some experience. I know that it's quite common for like a team to be completely wet lab and they don't really have anyone computational. So maybe, try and convince your PI to bring someone in to collaborate, to help with the computational bits, to try and help you. So yeah.
There's another question here, just to emphasize how this is a common feeling from Renan Simons. Really good at presentation, but any tips on how to balance what we think makes sense and what's really going on in the samples? So I guess you kind of answer that, right? I mean,
yeah.
You can just hope, but there are a lot of tools that you can go about. And
yeah, definitely. And try to have someone who maybe like has a bit more experience, and and always, I would say, err on the side of caution. Always err on the side of caution. My PI says I'm pessimistic every time. He's like, "Oh, you're so pessimistic." I'm like, "No, I just don't want to publish crap. Sorry." But just err on the side of caution. Actually, another good thing is try a few different tools. So there's, for example, for something like clustering, there's a bunch of different tools that, if your data isn't too large, you can use. Or, even for dimensionality reduction, look at your PCs, and stuff like that. Like, basically try and look at your data in multiple directions. And if it's consistent, it's probably true.
That's great advice, since there's so many tools and so many ways of interpreting your data. If you don't stay a little bit on the very cautious side, there's a lot that can go wrong.
Yeah.
Next question from Karina Oliveira. Great presentation. Is there any way you can evaluate which numbers for number of neighbors and minimum distance would perform better for your data?
So kind of touched upon it. It's really hard to know. Essentially, just try a few different things, see how it looks, see if it makes sense. Generally, nobody's really gonna like if you publish a paper, the reviewers aren't gonna be like, your number of neighbors is way too small. This is this is not reasonable or anything like that. So I mean, it's actually surprisingly, when you try these different parameters, the data generally like looks subtly different, but not necessarily too different. There is such thing as going too low, I suppose, but yeah, like it's just basically, you know, try a few different things, see what it looks like, and yeah, see if biologically it makes sense based on based on what you know about your own data, which, you know, you will know more than me.
That's a great answer. We still have a little bit of time, so maybe one or two more questions. So
Yeah,
Carla
no problem.
Vargas,
Yeah.
thanks for the talk. What type of annotation do you think is better? Would you use dataset from your own lab?
No, not necessarily. I don't think there is such thing as better. I think both sort of manual annotation and automated annotation, if you can use, you should use both. I think the most important thing, if you are like dead set on using markers, is don't just pick one marker and be like, that's my marker, it fits my data, therefore it's true. Make sure that you know, you have a good number and that they're all actually consistent. In terms of whether the data set used for automated annotation should be from your own lab, definitely not. I mean, we've used mouse data from a paper from 2017. We did our work in 2021, completely different lab, different institute. Actually, I think different technology. I think that data, oh, no, it was 10x, it wasn't SmartSeq. But we've actually also done it with SmartSeq data, and it worked reasonably well. So use any sort of data set. You know, if you're working with cells where there's like a ton of data, use a few different data sets. You know, if you work on something like, you know, blood cells, like PBMCs, oh, there's a wealth of data available out there. You can use like 6 different data sets to try and annotate your data and check whether it's consistent. And it's really cool when it is, because you're like, okay, like this makes sense. This is actually what we're seeing, like it's true. So yeah, like don't think anything's better, can definitely use any kind of data.
That's a perfect way of putting it. And the final one, a little bit related to that, because is asking about data reduction process.
Yeah.
You showed different tools for each one of the steps. Could you suggest a combination of tools that enables rigorous data reduction?
Yeah, so kind of each of those steps are like, they're kind of sequential steps. So, you know, you will always, well, you don't actually have to, but pretty much everybody will always select highly variable genes in the data, because single cell RNA-seq data inherently is very sparse. So it has lots of zeros and lots of basically dropouts. So you have genes that are like not expressed in any of the cells. So that's generally what everybody does. So you basically just make the data set a little bit less noisy by picking the variable genes, and then you do PCA with what you have. And then once you have the PCA, then you run either UMAP or t-SNE, whatever is your favorite, or both. On it. So all of those things are, you know, that's a sequential step of things that you do for your data to sort of arrive at the final embedding, UMAP embedding. So yeah.
I think someone here was talking about the fact that you said you could use different data sets, and even though different sequencing platforms, using different for the previous question, right?
Yeah. So yeah, I mean, I think I think it should be fine. I mean, it would be nice if like whatever you're comparing are both aligned to the same reference. So, you know, if your query data set is GRCH37, then your reference should also probably be GRCH37. But in general, like, I mean, it works better if it's the same platform. Like I found that, like, you know, if I'm doing automated annotation, sort of 10x versus 10x works a little bit better, but it's not impossible. Like you can still do it. It's, you know, it still works.
Gerda, thank you so much, and so much for staying with us for the questions and answers. It was really good. Please stay with us if you can, if you have some time. But we're right on time to move on to our next speaker. Thank you very much.
All right, thank you very much. Cheers.
So our next speaker is already here. I would like to introduce you to him. So Dr. Milos Nikolic studied mathematics with an emphasis on computer science at the Faculty of Mathematics from the University of Belgrade, Serbia. In 2013, he moved to Cologne, Germany for a doctoral position at the Medical Faculty, University of Cologne, aiming at better understanding the basis of gene regulation of common complex diseases, such as aging or orofacial clefts, by using computational analysis of next-generation sequencing data. There he was a shared postdoctoral student in two research groups, Professor Argyris Pentonis from Systems Biology of Chromatin and Professor Alvaro Radaglesias from Developmental Biology. In 2017, Dr. Nikolic joined the group of Professor Martin Pfeiffer in computational cancer genomics as a postdoctoral scientist, where he's currently investigating mechanisms of therapy resistance in small cell lung cancer by developing computational tools and novel machine learning methods on clinical data. Welcome, Milos. Thank you.
Thank you very much, Patricia, for this nice introduction. And thank you for inviting me here. So, yes, let me just try and share the screen. Okay, it should be fine now.
Yes.
Yes, so as you already heard, so yes, it's my name is Milos Nikolic. So I'm currently in Cologne doing a postdoctoral studies, as you wish. And yes, we are mostly investigating cancer in our groups, and more precisely, we're interested in resistance that occurs after therapy. So for that, single cell data is really ideal to look into details, what's going on, and to investigate cancer evolution. So today I'm going to present you a tool that we developed within the group. There is a small typo in the schedule, so it should be single cell RNA-seq, not the RNA-seq, but that was my mistake. So of course the whole workshop is about single cell, so it should be fine. Yeah, so the title is the M3K. It's a tool that is used to efficiently pre-process data. So everything that Gerda was telling you in the previous session, or well, the presentation, that comes after my part, let's say. So here the tool is intended for pre-processing data, so it's more like a rough pre-processing, and then you can use other tools on top of that. But you will hear very soon about it. So let's begin. Oops. All right, so probably most of you by now are familiar how the single-cell RNA-seq experiment works, but let's just quickly go through it. So you start with these 10x barcoded gel beads, and which contain barcodes and enzymes for reverse transcription. So then you put them in an instrument from 10x Genomics, and you try to pair each gel bead with one cell, and then you encapsulate them into an oil droplet. So hopefully you would want to see one cell with one set of these reagents and barcodes, which, as you can see from the picture, doesn't always happen. So sometimes these droplets are empty, they don't contain any cells, or they can contain more than one cell, so which is also a problem. So then in the next step, you collect these droplets, then these reactions, so for reverse transcription occur, and then you get the cDNA, which is then barcoded. You remove the oil and you get your sort of a library, or well, you get the library after you after you multiply, basically you get the library that is then sequenced, and yeah, you send it to sequencing, and then you get your data. So we were lucky in our case in Cologne, thanks to Roman Thomas and other initiators, to get our own instruments. So this gave us a certain edge to work with these types of experiments. So here is the workflow of the tool. It has 7 modules, and I'm going to briefly explain each one of these modules so that you have a feeling how the data is then analyzed, and it also allows you flexibility. You can adjust stuff by using parameters and so on, and then you can use tools for the downstream analysis at your wish, so depending on the research question you have and so on. So let's begin. So the first module is a very basic quality control module. It uses already made tool, which is called FastQC. Probably many of you are familiar with this tool. It's highly popular and efficient, and it's used not only for single cell, but for any type of next generation sequencing. So here I don't have any example of it, but you get statistics about the reads, sequenced reads, such as quality of the reads, if you have overrepresented sequences, and so on. So this is incorporated as an external tool, but it's very handy to take a look at the data if you don't know really what you should be looking at. And then here you have an overview. What do we really get from single cell RNA-seq data? So we have two reads, or basically we have forward and reverse strands, so reads from these strands. In read 1, here many things are depicted, but in the end you're basically left with just these 2 things, and that's the 10x barcode, which denotes the cell itself, and it has 16 nucleotides in length. And then on the next one is the UMI, unique molecular identifier, so which denotes different transcripts within the cell. Depending on the chemistry that you're using, so you can have UMIs of length of 10 nucleotides or 12 nucleotides. So version 2 chemistry is the older one. I think they don't even produce the kits anymore, but still you have many data sets available and published already, and there's nothing wrong with it, but you should be aware of this. So which chemistry was used in your experiment. So there basically is no coding sequence here that you can use, but because it's located here in the read 2. So there is the actual sequence that you're interested in that contains all the information. That's why I did the experiment. And so now we have a slight problem, because then in order to map the data, you will see in the next steps, we need to put the barcode and the UMI into read names here. And there are also a few steps that need to be done before. So apart from the FastQC part for initial quality control, there is also a couple of programs that we developed ourselves. So the first one is just to count the nucleotides in the UMI sequence. We had, from our previous experience, problems with this part because we were getting these sequences ending with many N's, N nucleotides in the end. And because the UMIs don't have a reference set of UMIs available, these should be completely random sequences. We did not have any ways to correct these sequences. In contrast, barcodes have a reference set of barcodes, and you can use them to correct barcodes, which I will also explain in the one of the next slides. So what was going on in our case, we would have in the final positions around 10th or 12th position, we would have here a very sharp peak in ends, and this would signalize that something actually went wrong in the sequencing facility. Then the next part is that we implemented here is that we want to check if the cells, or if a small fraction of cells, and by small I mean 10,000 cells, that's the cutoff here. Sorry, I lost my mouse. Okay, that's the cutoff here. So we want to make sure that the majority of these cells got the most of the reads. So when you perform an experiment, there is a theoretical limit that you can only get 10,000 cells from your experiment. But when doing the experiment, you actually put far more barcodes. So you mark, here you see 150,000 barcodes, and these numbers are even higher. But most of the cells, or these barcodes, will just contain a few reads or a handful of reads, and we will just discard them because these represent dead cells or contaminations of some sort, or empty droplets. So we don't care about these reads. But we also want to make sure that these top 10,000 cells should contain most of the reads from the sequencing. And in contrast, here is another example where this is not the case. So we actually are not sure why this happens, but sometimes you have these samples that really the distribution of reads, it's more spread, let's say. So it's more spread, and it's not really shared only between top 10,000. And this is also a cutoff that one can change using a parameter, but we usually use 10,000 because, as I said, that's the theoretical limit of the experiment. So this is the first module. We just look for quality checkups, and then we move on. The next step is to split data into different, into chunks. Why? I mean, it's just because of the efficiency. We do this on using high performance computer, and then we can just speed up the process itself. And here in this, let's say, picture, you can just see example of reads. I'm sure you're all familiar with it, but let's just go. So one read has these 4 lines. You always have a read name, then the sequence itself that we're interested in. And you have this plus, and then the last row is the quality of the sequence. The F is the highest. So this is just to give you an illustration. But so what we do is just split the data, and then what we need to do, we need to extract the barcodes and UMIs, as I previously mentioned. So from read 1, and then eventually put them into read 2. But in between, there is also a small step, which is to correct the barcodes. Why do we correct? So it's very simple. Whatever you sequence is prone to sequencing errors. And since you also sequence barcodes, they can contain errors as well. So we can correct barcodes because, as I said, we have reference set of barcodes, so we know how they should look like. But we can only correct those that have one mismatch with the reference barcode. So if they have one nucleotide that is different than in the reference barcode. And what we also do, we encode the barcode before putting it into read 2, just to save some memory space. So here you can see it's 16 bytes, and then when you encode it into a number, you just use 8 bytes. But this is mostly because we produce many temporary files, which are anyways deleted in the end. but not because the memory issues. Because of the memory issues, you increase the efficiency of the software, because it's much more efficient to compare numbers than strings, basically. So that's the basic idea. And so, as I said, we want to make sure that we can that we try at least to correct barcodes with one mismatch, and then eventually we want to put them into a read too. So basically, we want to add them here into the read name. So let's just briefly see how this works. So don't be scared. It's very basic, and I would like you just to grasp the idea how this is done. I'm sure it's nothing complicated. So you have one barcode here as an example, and then we split it into 4 mers. So 4 mers are just, you know, strings with 4 nucleotides, like it's shown here. And then each 4 mer is encoded into a number. So these are all then examples. How do these look like? And then each of these numbers are just kept in a hash table, which is basically a data structure through which you can look very fast. So it's very efficient in searching the data. And it's also that's why we use it. So each of these corresponding chunks, it's saved in the corresponding groups of variables. And we repeat this for all the reference barcodes. So then we go and compare with our barcodes from the data, from our sample. We want to see if each of these chunks is here contained in each of these groups. So for example, I can say, okay, I have the first 4 mer, I can find it here. The second one, I can also look and find it here. But for example, maybe this one cannot be found, and this one can be found. So in total, I have, let's say, 3 hits. And if I have at least 3 hits, that means that I potentially have a barcode with 1 mismatch. So it can have 1 mismatch here, or in any other position, or it can have more than 1. So it's between 1 and 4. So that's a potential candidate that we can try and correct. And then we look more closely and see if it's really 1 mismatch or it's more. If it's 1, we try to correct uniquely, and if not, we just discard these reads. And you will see in the end how does this look towards the end of presentation. The next step is to map to a reference genome, and for that we use a star liner. It's a very well established tool, and I'm sure you heard of it, and it's widely used for single cell RNA-seq data analysis. So here I just want to show you how it's illustrated, how the mapping process works. If that is also if you're very new to next generation sequencing, but if you did anything, any other type of experiment that you sequence, the principle should be the same, and you are probably familiar with this. So you start from the set of reads, and you have your reference genome from human or mouse or any other organism. So you want to put these pieces of sequences, you want to put them or map them back to the reference genome in the best possible way. And that's how you get these pile-ups. And here your sequences are very short, but in single-cell RNA-seq data and in other sequencing types, you have, of course, longer sequences. Here is, I think, 75 nucleotides is something that we are using, for example. But just for the illustration purposes on this example, these are very short. And then you try to find the best match on the reference genome. And then the next step is, once you map your reads to the genome, you want to see do these reads actually overlap some of your genes? So you want to quantify these reads on genes and you get these read counts. So you get counts per gene. That's the idea, and also within each cell. And in this case, we sort of implemented it ourselves, and we focus on 3 different types of cases, and I'm going to briefly also explain them. So this is the first example, and imagine that you have one coding sequence, gene or an exon, and then barcode 1 means these are all reads from the same cell. They have the same UMI, and you can see that really they overlap from beginning to the end. So that's the starting position. They're all the same. And this is very easy to determine, you know, which case is this. It's very simple. So these are duplicate reads. And you would usually just keep one of them because you get these reads when you amplified your initial sequences, basically, that you want to sequence. Then the second case is when you have reads from different cells and they don't really have the same starting position now, and the reads belong to different cells. So this is really okay. So they're all somehow different, and so they have different starting positions, they're different UMI numbers, and they belong to different cells. So that's great. These are all reads that we can use. And then there is this third case, which is a bit maybe weird. And here, the first 3 reads, they do belong to the same cell, but the other 2 reads belong to other 2 cells. So we're not focusing on them. Let's just focus on the first 3. Here you can see that the UMIs, so it should be the case that all the transcripts are different because they also have different starting positions. But according to this paper, you can have duplicate reads even though they don't really have the same starting position. So the starting position can vary. What they claim is up to 3 nucleotides, and they proved this experimentally also. So this is what we're applying here. So we consider these cases as potential duplicates. But then you might ask, okay, but they have different UMIs here. So what's the deal? Why do you think they are duplicates? We then, since can change one nucleotide, for example, in UMI1, and produce the UMI2 or UMI3, then we consider them, so that is something that is called the Hamming distance of 1. So if you can derive one of the UMIs from the other, and if they are on the same gene, we consider them as duplicate reads, and then we would remove them and just leave 1. And then the next step is to determine the number of viable cells. And here what we do, we look for the knee, basically. You want to, so the x-axis is the log scale. Sorry, I didn't mention this, and it's not written. And the y-axis as well. So it's log 10. So we here have the number of quantified reads per cell. And as I said, we look for the knee, and we use this just by determining derivatives of the curve. We can just determine this point. And this is done automatically, but also if you don't like this, you can set it up yourself. And if you use any of the downstream analysis tools like Seurat, they have additional checkups where they filter out other cells. So actually, I do like when I keep a bit more cells, and that, you know, in the downstream analysis, they are anyways going to be removed. So this is one of the final parts that the pipeline does. But before we go to the final thing that the pipeline is doing, I'm just going to make a small digression and explain you briefly what we're doing so that then you understand why we did this last step and why this is also needed maybe to other groups as well. And so we're working on small cell lung cancer, which is highly aggressive lung carcinoma, and that comprises around 15 to 20% of lung cancer cases. So patients often respond very well to the initial therapy, but then later on they relapse and they basically die. So the 5-year survival rate is only around 6%. And why is it aggressive? It's aggressive because it's highly metastatic. So basically that's what it means. And this is because of the high abundance of the circulating tumor cells present in blood of the patients. So these tumor cells circulate and then they enable metastasis. Or tumor growth in other organs. And here you can see examples where it usually metastasizes from primary tumor. And so what we do, we have data from these patients. And by data, I mean we have not only tissue from tumors, but also we have these circulating tumor cells which you can purify from the blood. So using these steps, which I'm not really familiar with, but you can extract these circulating tumor cells and then you can just inject them into mice. So these mice basically don't have any immunity. And that's why eventually tumor starts to grow in them. And we have many of these mouse models, and they're really great to explore all these things. But currently we're interested in genomics and gene expression. I also didn't mention here, you can also grow generations. You can hit them with different types of therapy or irradiation or chemotherapy. And this is really great, great model to investigate cancer. And it already has been proved that tumors that grow in mice, that are seeded in this way in mice, they're comparable to original tumors from patients. So you can use this data and be sure that what you're getting here is valid. It's comparable to real case scenario. And that's why you can imagine that once the mice are sacrificed and you extract the tumor to perform the single cell RNA-seq experiment, that is why you can have some sort of contamination, right? So you can also take some mouse cells and perform the experiment. So what we're trying to do in this final part, and this part actually comes just before you determine the number of viable cells. So we're trying to remove these host cells or contaminated cells, how we call them. And for that, we just have a very simple scoring method. And we obtain this score by mapping not only to human genome, if you remember from previous slides, but we also map it to mice. And then we have these counts per cell, and then we can derive this score. So it's the number of reads that was mapped to human divided by the sum of the reads mapped to both human and separately to mouse. And when you plot this in a sorted, you sort them by the number of, by this score, and then you can also derive this cutoff automatically by just smoothing the curves and finding derivatives. So you want to find this turning point again, or a knee, and you hope that the other cells are going to be cells from your model organism or from humans, so that you can use. Of course, here you will have also some dead cells or maybe even doublets or empty droplets. So it's not bad to remove them in this part, but when you determine viable cells, you also remove additional group of cells. And to see what we're getting basically when we remove, so when we focus on the removed cells from here, so what you see here, it's a group, I think it's 28 samples. So these are from these PTX mouse models. And then when you map them to human, you can see that they cannot be determined where do they belong, but in mouse they have many functional notations here. So it seems that the method works, but I will also tell you in the end that we plan to verify it in a different way, experimentally as well. But before, I would like to show you how the final report of our tool looks like. And I have to thank to Carlos Piage Jr., who was the main actor actually in this part, because he mainly or mostly developed it by himself. So thank Carlos again. I have to thank him again. This is really a great addition. So what you have here is one sample. It doesn't matter the ID, and you have several tabs here, and let's just see how it looks like. So in the overview, you can see the total number of reads, and that's what you get from the sequencing facility, and this is the final number of cells that you get in the end after putting this cutoff. So these should be the viable cells that you can then plug into other tools as well. So here you have input parameters. I'm not gonna go through them. Actually, it's not so important, but the tool offers a lot of flexibility, and all steps are more or less, they're basically transparent, so one can see what was done and adjust things if needed. And so that's just in this overview page. So then the next part is the quality control. You saw that already briefly, and so this is clear. So we want to make sure, as I said, that most of the distribution falls here, so the highest number within the top number of cells, and that here we don't have any problems from the sequencing facility. Then in the results page, you can see first, so these are again log scales, so both X and Y. So you see in this full line, that's the number of quantified reads, and the dotted line denotes the number of reads. So before you sort of try to overlap them with genes. So of course, when you overlap them, not all of them are going to fall on the genes. So that's why you have lower amount. Then you also have the cutoff, and here you have the actual numbers. So you started with this amount of reads, and then in the top 10,000 reads, we have 89% of the of the data. It's here. So and then the others 11%, they're just discarded. And so here, actually, in this case, the duplicate rate is a bit high. Should be lower usually, but I think the sample was sequenced deeply, or more deep than usually. So then you also accumulate the number of duplicates as well. Then you have very small amounts of these other groups of reads that were removed for various reasons, and we went through them. And here you have the barcode information. So you can see how many reads had problems with barcodes. So it's usually around 3 or 4% from our experience. And usually we can correct around 30% of these reads. So which is more or less what you see here. I think this number is slightly higher. As I said, if it's sequenced deeper than we usually do it, then you correct one barcode and you have many reads that are duplicates in this barcode, and this adds to this number. So because of this depth of the sequencing, I believe that these numbers are slightly higher in this case. So it's not ideal, but it's a really good sample, I would say. And then the not corrected barcodes in the end that we don't use basically. And here you have distributions, how the reads fall onto genes, and how the distribution of the UMIs basically in your sample. And then very important thing is mitochondrial genes. You want to make sure to have them on a low level of mitochondrial genes, because basically if you have higher levels, this means that your cell is dying, and that's not good. So if you would have higher numbers here, you would probably remove these cells completely. So, and this you can do, for example, with Seurat easily. But in this case, it looks really good. And then you also you can just compare the number of UMIs versus the number of genes to see if there are some, there's something weird or that everything is fine. And then the final part is just this, in case you have these PTX mouse models, if you have some sort of contamination problems, in this case, it's, a very small amount that was removed. And yes, so what we so far did to verify that our software is working, we mixed cells from two different types. So these are these PDX mouse models, and then we have the cancer cell line. And we just mix them together. So we sequence them separately, and then we mix them and analyze them as one sample. Or actually, I don't remember if we did it like this or we analyzed them separately and then combined the data. But in any case, so one has to take care if you normalize the data or not before merging the data sets. That's what's important. And our pipeline gives you raw data counts, so you can do whatever you like afterwards, any type of normalization, either per library size or some other ways. If you want, you can also do this batch correction and so on. But so here you can see, so what we wanted to see here is if the reads are actually mapped to correct genes, and we mix these two different basically cancer cells, because the PDX mouse models, so these should contain neuroendocrine markers, because small cell lung cancer is a neuroendocrine type of cancer. And for that, you can see these three genes, and they are all on, so they are all expressed. But in contrast, so this is the PC9 cluster, which has the EGFR, and the other, in other cases, you don't see it in the other cluster, basically. So it's also good that we more or less have two clusters. This one is somehow from this one, but it's separated. Although, I mean, you plot this with t-SNE, and in t-SNE, you know that distances don't really matter. So you can have very distant groups, but it doesn't tell you anything about the actually physical distance between them. So it's not a measure. So it looks good, and we have another sample that was combined with PC9 cells, and it's also looking good. It's, if you have these two groups, in one it's expressed the gene, in the other it's not, and so someone asked in the previous lecture, how do I know that I'm not, doing the analysis in the wrong direction? So this is one of the ways. So you have to be creative and see if the data makes sense. If you have certain cell types you want to check for markers, and you also want to check for markers which shouldn't be there. And this is, one exercise like that so you could perhaps apply that. And Yes, so that's one part of the verifications for the overall tool. And for future work, we are actually finalizing the program optimization. And we want to compare it with Cell Ranger, how it performs. We think it's going to perform better also in terms of time. But also, I mean, nowadays, Cell Ranger has very efficient pipeline as well. But we think we can beat that. But you know, I can't show you anything at the moment. And there is this one experimental validation that is ongoing, which is to mix cells and perform the 10x experiment, so for single-cell RNA-seq. And this cell mixture then to sequence it and hopefully to verify this method also experimentally, that you would want to hope that these mouse cells are going to fall here in this tail, that you would just get rid of them, and that what you're left with, you can be certain that these are human cells. And we would of course choose different ratios, 5%, 10%, or 20%, or a couple of these experiments to verify the method. And in the end, so in summary, so you can see that this tool can, extract the barcodes, correct them, map reads, then provide you with raw counts from the gene expression per cells. You can remove duplicates, remove contamination, and determine the number of viable cells in the end, which is, all these steps are somehow standardized more or less. So that's what the other tools are doing. For us, we always like to implement things ourselves so that we're really certain that we know what's going on. We cannot, continue to investigate cancer by just guessing, or just assuming that everything that was so far done was correct. And so you can, as I said, you can adjust parameters. And currently the GitHub the project is in private mode, but in the following weeks, we hope to make it public. So if someone is interested, please contact me per email, and let's just talk and see if you're interested, and we can support you probably in that. And in the end, I would like to thank to Julie and Clydeson, Marcel, Carlos, Victor, Johannes, and Lucas, and my bosses, group leaders, Martin and Roman. Yes, so now I'm ready to take any questions that you might have. So thank you for your attention.
Thank you so much, Miles, for this great talk. We do have a few questions. I believe some of them you have already answered, but or talked a little bit about them, but people are interested in knowing if the tool is available.
Yes.
And
so it's currently in this sort of private mode. For example, Carlos will try it out in his center in Brazil, where he's working now. So we want to test the tool a bit more, although in terms of code and implementation, everything is working. We've been using it for a while now. We have plenty of samples here, but please be patient a bit more. We also are in a process of writing a paper. It's going to be very soon, but we need to finalize this analysis first. And yes, that's, but if you're interested, just contact me and I can talk to my bosses, and let's see what we can do. So it should be on very soon.
Mm-hmm. The link for GitHub, can they run it from there? Is it available there?
So when you get it from GitHub, you would need to copy the files. So currently it's not available because it's still in a private mode. It's not public. So, but when you get it, you would need to copy the files to your high performance computer, so to a cluster. And then from there, you need to set up some paths to these external tools, or you might want to install them first, but that's going to be in the manual. So it should be very easy to install it, and then you just need to run command. It's just one command for this whole sample. All the 7 modules are going to be submitted as separate jobs, and they can be also done in parallel and so on.
Just a reminder for everybody that the talk is going to be available later in YouTube. So both the link and your email, they can find it there afterwards, right, to get in touch with you. One question here about the tool again, is specifically about availability, if it is just for 10x technology or other technologies are supported too.
Currently, it's for 10x technology because we use this reference set of barcodes, as I mentioned, and if you would use it for from other technologies, we would need to test this first. It could be it would be possible probably, but one would have to go through each step and verify that this is really working. So I wouldn't recommend just directly trying it, because then it's not going to work. But if you're interested and so on, we can talk about it and see what we can do.
Okay. Another question here, you kind of talked about it a little bit in the end. So great presentation. I'm a complete beginner in single-cell RNA-seq. I understand that M3K corrects the FASTA-Q files before and also creates the count matrix. So can it replace Cell Ranger? One of the main disadvantages of Cell Ranger is the computational power needed to do the analysis. The computational requirements for M3K are as high as, they be as high as Cell Ranger? You kind of talked a little bit now in the end, right?
Yes. So that's the general idea. So when we started working on these data sets, there were just few initial versions of Cell Ranger. The analysis took, I think, 2 days. And we also didn't really understand what was going on in each step of the process. So that's why we decided to do things ourselves. In the meanwhile, they did speed up quite a lot compared to the initial versions, but still, for example, they don't offer this support for the Slurm, this scheduler on the cluster, although people reported that it's working and that it can be installed. If you manage to do it, it's a bit of a hassle, but if you manage to do it, then it's sort of faster than in this other way that maybe most of the people are using. So, but the idea is to just, use the M3K instead of the Cell Ranger. That was our goal, or at least to offer an alternative, because I think Cell Ranger is the leading tool that is used for that. And yes, I don't know if this has answered.
Yeah, it does. And if people would be willing to test it, to compare, are you looking for collaborators? Would you like people to be helping out with that? How does that go? Because the tool is still being tested, right?
Yes, so we will test it, and we will do it in a way that on the cluster you can dedicate certain resources really for both tools, and you can then just measure the time. So that would be fair comparison, so that each tool has the same amount of resources that they can distribute how they like, but we have a plan to do this, and we can do it locally because we have so many samples. We also have many publicly available data sets are there. So, but of course, you're free to try it yourself and see if this makes sense for you. If this is really comparable to Cell Ranger. I'm sure it's not going to be exactly the same, because there are a few things that Cell Ranger does differently. And one of them is that so when you map the reads to the genome, you want to make sure that you have only these primary unique alignments, so that your read maps really to just one position, not to multiple, and you keep only those reads. So what they do in Cell Ranger, I'm not sure if they changed it in the newer versions, but I think they didn't. So they somehow impute that some reads are primary alignments, even though they map to multiple places. So, and for us, this is somehow, you know, it's artificial. So we want to, you know, we don't care if we remove a bit more reads, but what is left, to be sure that it's, you know, these are proper reads, then to try to, you know, somehow maximize the number of obtained reads in this way at least. But I can understand them. It's also, I mean, they produce the technology and they also produce the tool, and you need to sell the technology as well. I mean, you need to claim that you get many reads, although this is a very small fraction. We're talking about very small variations, but you know, that would be one example why I would expect it to be different.
But would you say that your main motivation to develop this tool was the computational time that it takes, or the fact that you don't really know exactly how they are running it?
I mean, I think we understood eventually how they do it because we were in contact with them, but also throughout the history of the groups where I am currently, it's imperative to develop our own tools. So we do step by step. It takes longer, but eventually we are certain of in our results that things make sense, and you know, improvement of the cancer treatment is the ultimate goal, and something that is transferable to patients. So that's the idea. So it's, you know, it's all these reasons. It's the tool is not really flexible. It's not easy to adjust it, but you know, now when I develop this tool, I can adjust whatever I like. I know the code. I know every bits and pieces of it, and so that's you know, it offers us flexibility. We might need to adjust for some cases.
Possibly related to that, when you talked about barcode correction, you implied that there is an impact for the downstream analysis because you're collecting more reads, right, for specific groups. Do you have any experience on how much this impact, or if you can tell from your raw data, how much this will impact the results downstream from that?
Yeah, so it depends. I don't know if it's meant on for barcodes or the UMIs, but in both cases, if you saw in the report, these are all very small amounts. So of course, the barcode correction of the actual cells, I mean, I think it's important to do this, and this is also done in Cell Ranger. But with these UMIs that we sort of we can conclude that that they have sequencing errors in them, and then we remove them. It's a very, very small fraction of them. It's indeed, I mean, we could just ignore this, but since we knew about it, we just decided to do it.
All right, and the final question, great talk. Which quantification program or algorithm do you use?
So that's the thing that we did ourselves. So we implemented this in C++, and we have our own program. So you just read your mapped reads from a SEM or BAM or SEM file, and then you also read from a GTF file. Currently, we support this AG19 and MM10 for mouse, but we will provide in the GitHub these GTFs for also the newest human genome. And yeah, so we implemented it ourselves. That's why there was this verification where we mixed the PDX cells with PC9 cells and looked at these genes just to see if we get quantified the things that we need, that we should have. So that was a sort of a checkup. So there is no tool, we did do it ourselves. So it's not a pipeline we have. So the core programs are developed in C++ for the efficiency, and they're parallelized, and also you split the sample into smaller chunks. So you get this, it's sort of like a hybrid optimization. So it's.
Yeah, I think this answers the questions. Well, Milos, thank you so much for being here. We do have Carlos Biaggi with us in the team for the second week as one of the trainers. So I believe he will talk about this a little bit more, or our students are going to have some more questions about it, and we may reach out to get some ideas from you. Thank you very much.
Thank you, and I just have to thank the audience again for staying with me. So enjoy the rest of the workshop.
Welcome to the afternoon session of this second day of our workshop. It is my pleasure to introduce Robert Amezquita, our next speaker. Robert Amezquita is a postdoctoral fellow at the Immunotherapy Integrated Research Center, YYRC, at Fred Hutchinson Cancer Center, under the mentorship of Rafael Guetardo. His current research focuses on utilizing computational approaches, leveraging transcriptional and epigenomic profiling at single-cell resolution to understand how novel anti-cancer therapeutics, ranging from small molecule therapies to biologics, such as CAR T cells, may affect immune response dynamics. Extending from his graduate work at Yale's Department of Immunology, Robert's research aims to better understand the process of immune cell differentiation under the duress of cancer as a means to inform future immunotherapies. To accomplish this, Robert works collaboratively across the Fred Hutchinson YYRC with experimental collaborators, extensively utilizing R and bioconductor for data analysis, and he will be talking to us about trajectory analysis. So please feel welcome, Robert. You have our undivided attention.
Can you hear? Hello? Can you hear me? Voice?
Yeah, we can hear you.
Oh, okay, great. Yeah, hi, thanks for the great introduction. And yeah, it's my honor to be presenting today, trajectory analysis, giving an overview and some theory behind it. And yeah, I think this is a really interesting topic because the single cell RNA-seq landscape has really unlocked a lot of amazing sorts of techniques that we can apply to better understand various different processes. So I'm excited to be presenting this topic here. So the outline of my talk today is going to be going over what exactly is a trajectory when we use this term, so relating it back to the biology, experiments, and the meaning of trajectory analysis. And then I'll just give a really brief overview of available methods from a recent review, a bird's eye view, if you will. And then we'll dig into the methodology framework. So we'll talk about kind of the contours of what a general approach to trajectory analysis entails. And then finally, we'll go through an example implementation where we actually walk through and deal with some of the limitations and constraints of trajectory analysis in a stepwise fashion, similar to how you might do it in your own analyses. So yeah, so let's get into it. So what is trajectory analysis exactly? So when we say trajectory analysis, what we're referring to is mapping onto a given data set. In this case, single cells are what we're going to be working with, but really, I mean, just single samples of some sort, biological units. I'll be referring to single cells because the predominant mode that we're going to be applying trajectory analysis is in single cell RNA-seq and other sorts of single, site-seq and whatnot. But so trajectory analysis aims to map cells onto some biological process, and thereby, gives these cells a relationship to one another by just being on a similar axis, a lineage, if you will. And so by performing trajectory analysis, we can map these cells, these data sets, to a variety of different processes once we have this new axis. So for example, trajectory analysis, immediately, one of the first things that comes to mind is mapping to differentiation processes. So when we look at, we see the early roots, which are the undifferentiated, stem cell-like cells, all the way to terminal cells. We can very confidently say that we're exhibiting, or we're seeing a differentiation process. But of course, there's other sorts of types of trajectories that we may observe: cell cycle, circadian processes, metabolism, disease progression, I think, is one especially of interest. that you might be looking at how cells change over time with respect to a given disease phenotype, homeostatic maintenance, and of course, signal response dynamics. So when you apply some perturbation, what happens in the time points thereafter? And so trajectory analysis, by placing cells along some axis, trajectory, or pseudo time is one term that you may hear. I'll briefly give a note about that. But once we place these cells into this imaginary axis, we can start to see various different processes much more clearly and the relationship to gene expression patterns. And so what are the types of motifs that we might see in trajectory analysis? What exists, and what do we think we exists in nature? What do we see in nature? So of course, one of the first ones is cycles. So here I'm showing the TCA cycle, of a motif we all learn when we go over metabolism. But of course, we also see it in the case of self-renewal of cells, for example. And so these sorts of closed loops are one such motif. And then we have linear sorts of motifs. So this is a one-directional sort of process that goes in just a line. So it's in a stepwise fashion. So this may be a process of differentiation, for example, linear differentiation. And then there's forking sorts of motifs. And so these forks here, we start to get a little bit more complicated in that a given node or state can give rise to multiple states. So one that I'm personally very familiar with is the T helper cell differentiation process. So depending on the context of the infection, we get different types of T helper cells. So viral versus bacterial infections will give rise to different T helper lineages. And so by the fact that we have this sort of multifurcation process, this sort of motif somewhat complicates the analysis, and is something that methods really need to adapt to. And then finally, a tree is something where we see multiple levels of bifurcation or multiplication. And for example, charting out the hematopoietic stem cell differentiation from the earliest points all the way to the terminal sorts of cell types that we observe is certainly one that contains all of these different motifs. And so drawing trajectories is going to be an exercise in picking out one of these that we want to study and seeing how we might combine these all in service of trying to interpret the underlying biological process that is driving the cell's gene expression patterns. And so one question we might ask is, how exactly do we place cells along these axes? And so the way we describe, so we'll go into the how in a bit, but so what are the coordinates exactly that we're going to be using for cells, right? So the units of trajectory, so it's just an abstract concept in that a given value for a trajectory is not going to necessarily map to a specific time or day or whatnot. It's really just this arbitrary sort of axis where we have a fractional unit of a given calculated trajectory, and the units are based on the relative distance from a root node, so the very first node in the trajectory, and the distance away from that. And so the only way we can really get at this is by drawing a single line. And so there may be actually, as we see here, multiple lines that we can draw from root to terminal nodes where we can place a cell onto an axis. And so all of these, again, are arbitrary. Even within a given analysis, pseudo-time values from one lineage to another lineage may not be directly comparable. And so we just have to be cautious about the interpretation, not over-interpret the actual units of the values of the trajectory. So, but nonetheless, we could start to map it to real time processes temporal processes or time points, assuming we have, known data sets, known data where we have that sort of label. So I want to take a brief pause and I'm trying to avoid the term pseudo time for a reason here. And that reason is that I believe that pseudo time is a little bit of a misnomer because it really lends itself to giving us a bias in how we interpret the trajectory analysis results. And so what we need to be mindful of is that when we draw trajectories, we're really drawing axes, continuums that may, and the relative positions of these cells may or may not map to the actual underlying process that generates it. So for example, we can imagine a continuum of stress states and draw a line, between all the different cells in a given data set. But in actuality, it might be curves where there's a lot of other intermediate states that are not observed that are an essential part of the process, and we're just not drawing those links. We're not able to draw those links. So pseudotime is a little bit, unless the context is one of time explicitly, maybe we have labeled time points or whatnot. I'm trying to be as cautious as possible about using that specific term because we want to be able to be as unbiased in how we interpret these results as possible. Really, pseudotime is most applicable when we can infer the directionality of a process confidently. So experimental studies of temporal processes, of course, have been around for a long time. Time has always been something of fascination. I mean, everybody goes from baby to adult, that all involves time processes. And so how have we historically studied these, before the time of single-cell RNA-seq, is there are, would be in vitro experiments that use some sort of physical fractionation or chemical blockade to induce so that we get a population that is essentially purified for a given state. And so one of the big challenges is that this requires extensive prior knowledge in either what are the properties that would physically fractionate this given population of interest, or alternately, what would be the chemicals that kind of halt the cell cycle if you're studying that and get everybody on the same page, in G0, G1. And so so that particularly is difficult to get at experimentally. It's not tractable really for a lot of different sorts of biological processes that we're interested in. And so single cell time-lapse microscopy, besides the sort of limitation of having an in vitro system, is really limited in the number of cells observed and the number of features that we can quantitate. We really can only quantitate a handful of genes that are not going to obscure one another on a given on the light spectrum. They must be observable. So really, practically, we can really only get maybe up to 5, certainly 3. And so this is why single cell approaches are really exciting, because we're combining single cell approaches with high throughput sequencing and cutting edge computational methodologies. So single cell approaches, of course, gives us a very fine-grained view of biological processes. We can see it with unprecedented sort of resolution. And high throughput sequencing gives us a really rich feature space. We can quantitate over 30,000 features in the transcriptome of these cells. And these two allow us to really confidently, or not, maybe not confidently, but have a really refined and robust sort of model to draw of the inference of trajectories. So now one thing I do want to caution is that the data, the single cell RNA-seq data that we do acquire, a heterogeneous population in terms of the cells must be in the different stages of your process of interest in order to be studied. So of course, no single cell RNA-seq is necessarily going to bypass that requirement. For example, it may be a system where you have highly stable terminal states, and the intermediate states that would be necessary to observe in order to draw the link between those two terminal states simply are too rare. is a huge limitation. It's something to be very mindful of in, again, interpreting this data. But by and large, I think these, the combination of these three factors have really given us a new avenue for interpreting and looking at interesting biology. So my previous points might bring to mind, when should we perform trajectory analysis? What might we see, and how do we interpret it exactly? So I've been kind of harping on the interpretation aspect in particular. Well, as far as when should we perform it? So whenever there is some sort of observable temporal process, or sort of like some sort of biological process that has sort of like different stages and whatnot, of course, that's a natural, we should definitely try and apply trajectory analysis to have one more axis of interest that we can start to draw associations with regards to the gene expression profiles that allow us to interpret more about what's ongoing in the given sample. And so, and be able to go, from a single sample to having a lay of the land on a temporal sort of axis. So, I mean, in practicality, you can apply it whenever you want, but it's very easy to actually misapply it in many cases. And because a lot of these methods will draw lines no matter what, as you'll see in the example section that I'll walk through. And the issue is that if we do that, we're going to have a lot of different results. It's garbage in, garbage out. So again, we really need a continuum of states and to draw these reliable and robust trajectories. But notably, like I mentioned in the earlier example regarding two highly stable terminal states, the lack of an inferable trajectory or like sensical trajectory does not really preclude the lack of some process that links these states. We really need to recognize that distinct clusters may just be the endpoints of a trajectory with rare intermediates. And so, or condition-dependent sort of intermediates, like, for example, in the case of disease progression. And so, basing a trajectory analysis on granular fine-grained sort of clustering versus continuums, is another point, and I'll walk through sort of both approaches. And that's something that, once again, is left to the analyst based on what's more convenient, useful, and biologically sensible. Now, one more thing I want to touch upon is kind of a new, more recent development in the field of trajectory sort of analysis is RNA velocity. So RNA velocity is defined as being is defined by the ratio of splicing to spliced to unspliced RNA. And so this, taking these differences allows us to draw a vector of sorts per cell that, we assume is pointing to the future state of a cell. So we're looking at the mRNA pre-processing sort of step. And if we use that state and then look at a future state, we can do some math and infer what the future state of that unspliced versus spliced, what's the nearest sort of like approximation of the future of that state of that cell. We can start to draw these vectors per cell. And so it's a really fascinating sort of approach because it unlocks like a new facet of the data that we've had all along using, just by pre-processing the sequencing data, the raw sequencing data a little bit differently, adding on the spliced versus unspliced sort of count matrices, we can start to infer, using this sort of RNA velocity approach to draw these individual vectors. And so this gives us, if you've ever seen a wind map, it gives us that sort of picture, and we can lay out the wind map such that the arrows are all pointing kind of in a line. And would be a different and so today I'm going to focus on classical trajectory analysis. I'm not going to get into RNA velocity, but a lot of the concepts I'm presenting here in map nicely or are complementary to the analysis of RNA velocity. So briefly, let's talk about what are available methods. If you want to get out there and just try it, there are a whole host of methods that you can go to. So just historically, what were the first approaches to trajectory analysis? So first, we're kind of clustering techniques that were adapted for trajectory inference, things such as that were built on things like flow cytometry. So this was like SPADE, for example, from 2011. Monocle was one of the first ones to start to work on single-cell RNA-seq. Wanderlust, made a big advancement in introducing nonlinearity and incorporating continuity in terms of the methods, albeit it was really limited to single branch sort of linear trajectories. And then, other approaches have come out in the time since. For example, placing cells onto manifolds using diffusion maps is one such method that is not necessarily dependent on clustering, for example. And since 2015, there's been way so many, different sorts of software suites that work on trajectory analysis, work to perform trajectory analysis. So just to name a couple, Monocle 3 is one that's out there and very popular. There's bioconductor-based workflows. And naturally, there's many other software solutions, and even in different languages. So R and Python are the predominant ones, of course. And there's this really nice review by Salins from 2019, covers this this framework of benchmarking using gold standard data sets and simulated data sets, and looking at different facets of these different softwares. And this review is really comprehensive. It's over 70 methods that they cover. And what they've done here is quantitated in some fashion the overall, like the accuracy, the computational scalability, aka are you only able to work with hundreds of cells, or can you go up to thousands or tens of thousands, hundreds of thousands, and how stable are the results? If I run with slightly different parameters, am I going to get the same sort of results? And one of the aspects that I think is actually the most important one, if you get anything from this, is that all methods are very different. Sorry, maybe not very different, but they're all different in terms of their assumptions and constraints in which they, do the trajectory analysis sort of implementation. And what that leads us to is that each method is uniquely geared to handle specific trajectories, there are going to be methods that are really good for linear sorts of trajectory sort of ordering, and unable to handle multifurcation processes, bifurcation processes. So we want to pick the right tool for the job. There are going to be, of course, if we know nothing about the system, we can pick one of these methods that are very assumption-free and be able to try and infer what trajectories are existing in the data set. But naturally, this comes with its own caveats, as we're once again making a lot of assumptions. It's lacking these sorts of constraints. We're making a lot more assumptions, stronger assumptions in order to be able to actually analyze the data. So no one method is good at everything, and actually they create this really awesome workflow figure here that will help guide you in terms of what methods they believe are going to be the most performant for given sorts of expected topologies. So depending on what you know about the experimental system at hand. So this is just really great. If you go and look at anything from this paper, I think this workflow is definitely the way to start and pick out that one method to try out first. And yeah, so I'll be following along going into a deep dive, using this bioconductor-based sort of workflow approach that is described online in this Orchestrating Single Cell Analysis online resource chapter specifically devoted to trajectory analysis. So with that, we can get into the framework of the methodologies and just kind of understand the skeleton of a typical approach. All approaches, again, are different in their exact implementations, but this kind of gives us a overview of what are the broad steps that each method is performing. So, of course, the first step in any of these experiments is generating the raw data that we need. So doing the sequencing and creating the counts matrix, the raw counts matrix, or in the case of velocity, RNA velocity, spliced and unspliced matrices that we will begin to work with. And then after that, a very important topic that I'm not going to get into today is normalization and feature selection. Naturally, both of these are absolutely essential to preparing the data for subsequent steps, and actually have been found in some recent sort of benchmarking papers to be some of the most pivotal in skewing the results and their ability to skew the results, particularly normalization. So this normalization itself is a whole topic in and of itself, but just to make the point brief, that's something definitely to be very considerate of. Now, these normalized and feature selected sorts of matrices can then be count matrices or log count matrices or whatever, can be fed into approaches that perform dimension reduction. So this is really essential because working with 30,000 rows is really not computationally tractable. And furthermore, in the single cell RNA-seq, there's going to be there's just incredible sparsity in that there's a lot of non-detected events. And so what dimension reduction does for us is it reduces the data set down to a tractable number of dimensions. So we'll go from 30,000 features down to 50 features, let's say 50 coordinates, 50 axes of principal components. And what this effectively does is it denoises the data for us and also compresses the data for us, and does all this with minimal information loss. So we could include up to 200 or 500 even principal components and still not be anywhere near as computationally intensive as working with the raw data. So suffice it to say, of course, in practice, 20 to 50 components are more than enough for most data sets. So really, this is going to be what's important to recognize is that this representation is going to be the basis for learning the structure of the trajectory. So depending on what sorts of approaches you're applying at the dimension reduction step, if you're picking those out or if the method is doing that for you, this is basically going to be the framework that the method is going to be starting to link together the different either clusters or cells in this sort of relationship mapping fashion. And so from there, as I'm alluding to, we have to infer the structure that underlies this reduced dimensionality representation. So how do we start to draw edges between different communities and different cells? So this is predicated on calculating distances between cells by some choice metric, maybe Euclidean or some other choice metric of your choice. And furthermore, we then have to take these sorts of relationships, these distance sort of matrices, and form a graph, things such as a k nearest neighbor graph or shared nearest neighbor graph, whatever sort of graph you're interested in. Of course, forming that graph has its own parameterization. So, for example, how many neighbors are you going to be including? Are you going to set a cutoff distance to enforce the closer connections? Both of these have their own sort of parameterizations, and so the structure inference has a lot of different knobs that you can tune, and what that ends up leading to finally is having a skeleton onto which we can map individual cells to a trajectory axis. And so, for example, for linear trajectories, given a single axis, we're simply transposing each cell onto the nearest point along that axis, and that gives us the trajectory unit of a value associated with that cell. Now, again, one thing to recognize is that trajectories will have multiple different lineages or lines that we can map onto. So a cell may not map just onto one axis, but onto multiple axes, and so may be represented multiple times in a given trajectory analysis. And it's up to you up to the analyst to make a decision regarding that sort of presentation of the data. And then finally, we have downstream analysis. So of course, why do we do all this? We don't just do it for pretty pictures of the UMAP and TSNE, but we actually want to understand the relationship of driving gene expression processes. So what exactly is leading to this observed trajectory? And so utilizing this abstract trajectory unit assignment that's given per cell, we can start to map to, we have essentially a new axis that we can map other features onto and perform, even things such as simple things like linear regression to determine if there's a significant association between the trajectory and a given gene's expression profile. So as time increases, maybe the gene's expression is also increasing, and that would give us, and if that's observed and is statistically significant, may start to give us clues into underlying processes and things we may want to follow up on in future experiments. Of course, I mentioned that because it's really important to validate a lot of these results. Not done often enough, unfortunately. And of course, there's a lot to be said in the value of simply presenting these wonderful hypotheses as posed by trajectory analysis. But suffice it to say, these all come with the caveat that there should be alternate sort of orthogonal ways to validate these relationships. So let's go through an example implementation of how we might actually use trajectory analysis on the data set. So this is adapted from the Orchestrating Single Cell Analysis online resource. So once again, point you to bioconductors. So just look up on via Google, bioconductor OSCA book, and you'll see that you have this really large compendium of and trajectory analysis is just one of the chapters. There are other chapters devoted to normalization and feature selection and all sorts of different aspects of single-cell RNA-seq analysis in particular. And so we're going to only present, of course, the trajectory analysis sort of stuff, but there's so many more other things. I highly encourage you to check it out. So let's start with some data set. So this data set, you don't really need to know the specific data in this case, but suffice it to say there is a temporal process that we know is ongoing, and we get the single cell RNA-seq data set, we get the counts back, and we want to be able to map each cell onto this given temporal process because we don't have that label a priori. And so the sort of simplest approach that maps to kind of the historically the earliest sort of approaches is one of first clustering the data into discrete units. Now, note that clustering is really arbitrary in terms of the resolution and stuff, but we'll skip over that for a second. And for each cluster, we'll compute the centroid. So, we'll compute the cluster centroids and get things like an average expression profile, for example. So greatly simplifying the data set. And from there, we'll calculate the distance between the clusters, and we'll use those distances to form a minimum spanning tree. And so on the right-hand side, you'll see that the data is represented on a TSNE plot, a reduced dimensionality representation that takes our 50 principal components and distills it down into a human interpretable 2D representation. And we see that these edges here are drawn onto this TSNE and giving us the relationships between each of these clusters. So for example, we see that one terminal node is cluster 8 in gray on the bottom left, and that's its nearest neighbor is cluster 5. And then another thing to note is that we have a bifurcation process seemingly where we have cluster 3 either going to cluster 4 or going to cluster 2. And so again, something to note, this is just, we're making a lot of assumptions. We're making the assumption, for example, that say the Euclidean distance is the best way to represent the relationships, to draw out the relationships. But of course, there may be like non-linearities, or biology is always complicated. It may really be that cluster 1 is connected to cluster 4 or is connected to cluster 6. And based on this representation, we shouldn't overinterpret T-SNEs, but ostensibly it could be drawn any which way from going from cluster 1 to any of the other 4 clusters. That would be believable. So again, really important to check the stability of your results by tuning the parameters, just playing with that and seeing if you're getting similar results. And of course, validating via orthogonal measures, making sure it makes sense from kind of known a priori knowledge. We're using experimental labels. But so from there, from this minimum spanning tree that we've drawn, so these lines again on the right-hand side, we can map each individual cell to the closest edge. So essentially just take the distance of that cell, let's say even on the T-SNE, and just map it directly onto the closest portion of that edge. And going from the root, we will be able to calculate the relative distance or pseudo time or trajectory unit that corresponds. So one thing again to note is that T-SNE is just how this data is represented, and it happens to look really nice. But there are often times that trajectory analysis results do not map very neatly onto a human readable sort of like form, and complexities that don't really map nicely onto UMAP or T-SNE. So something to recognize that just because it might be messy doesn't mean it's not real, and so, or not a real result, at least computationally. So one thing you might be wondering, so we touched upon very briefly the potential for spurious connections, right? So you might have noticed that there's this tiny cluster, this pink cluster on the right-hand side, cluster 7. And it looks pretty different. It presents very differently. It's very, it's a very tight cluster. We might think that that's noise. It's maybe a low quality sort of cells that are clustering together. And so we're a little bit hesitant to draw a link between cluster 6 and 7. So we want some way to protect ourselves against spurious sort of connections that may form by happenstance. And these connections form because by definition, the minimum spanning tree is drawn on all clusters, regardless of, whether it looks like a good link or not. It's going to draw a link of some sort. And that's why I say, be careful in your application, because you're going to get a result of some sort. And the interpretation is where you're going to be wondering if it was the right application. And so how do we deal with spurious connections? So one way that we might think about is like, okay, well, what about like some sort of random cluster, like, that we can generate or something like that? Well, we can what's the probability of like linking to that versus linking to this specific cluster? So we can kind of do that actually by creating a artificial cluster, an outgroup that is equidistant to all the clusters. And so based on this outgroup, if we include that outgroup, we'll be able to basically filter out connections to clusters that seem to be a little bit too far to be real. So we might set some threshold at which point we are going to draw a link to the outgroup or prune edges and stuff and such. And so let's take, for example, this edge to cluster 7. On the right-hand side is the representation of the minimum spanning tree with the relative distances kind of shown. And we see that the jump from cluster 4 to cluster 7 is pretty large, represents the larger distance between them. And that there's an outgroup that exists between all of these clusters that are equidistant. So you can imagine that the edge length between 4 and that outgroup is much less than the distance between cluster 4 and cluster 7. And so we're going to draw that edge from cluster 4 to our outgroup. And that in and of itself is going to filter out the spurious connection. And then we can prune any edges that are connected to the outgroup and get rid of the outgroup entirely. And so we'll have a result that has dealt with this sort of spurious connection. Now, there's a lot of other methods, of course, that you might also think of, like sort of outlier sort of approaches, or even just quality control metrics that might catch such clusters that could be problematic to performing trajectory analysis. So always be sure that you're working with good data, as good of data as you can get, and as is possible. So now another issue is the problem of clustering. So I mentioned earlier that clustering, extremely arbitrary, right? We can go back to this picture and we could easily draw, increase the resolution and get a lot more clusters on this data set, right? So we can reduce the smears that we're seeing. So cluster 3, for example, might split out into 2 different clusters, same with cluster 2, all these. And that would create a totally different minimum spanning tree based on the clusters, right? We would get, like, you can imagine, a noisier minimum spanning tree. So for a given resolution, one issue that comes up that's related to this sort of resolution of the clustering is that we may have clusters that are really spread out. And these, when we calculate the centroid of these really spread out variable sort of communities of cells, the distance between those 2 clusters, even if those 2 clusters are interacting at some border that a clustering algorithm may be drawing a line at, then those clusters may be too far away from each other relative to some other random cluster. So how do we deal with these sorts of smears? How do we deal with this fact that we have very spread out clusters that may be touching in this high dimensional space, but are being excluded from connecting to one another on the basis of this very simple centroid to centroid connection? So one way we can deal with it is by using, again, the graph sort of approach, one approach, for example, the mutual nearest neighbors approach, to find regions where clusters are touching computationally, and use this connectivity measure to draw new edges between clusters. And what we're really going to be looking for is connections between the edges of the clusters to one another, and using that as the basis of the minimum spanning tree. And so on the right-hand side, again, you'll see that this ends up producing the same result, a very similar result, I should say, sorry, as the cluster-based minimum spanning tree. Now, we see that the connection from 1 is no longer to 3, but it's to 2. We can see that this juncture here, definitely we could see there being variability just on this result alone being combined with the previous analysis. So again, we'll want to dig into that more. Now, an alternate approach so now getting away from clustering. So let's think about a high dimensional space again, and that we have different communities of cells in this high dimensional space. Well, maybe we can just draw a line that connects all of these different communities in this high dimensional space. And so that's kind of the very simplified idea behind principal curves. So one way to think about it is that it's the extension of the principal component analysis to accommodate the first axis that explains the most variation. The first axis ends up bending, being able to account for more nonlinear sort of variation present in the data. And so this is really nice because it's super simple. We're just again, nonlinear generalization of the PCA, but you'll see here, however, that it has a very different result from the previous ones, now connecting cluster 1 to cluster 4 more closely than clusters 3 or 2. So this may be questionable because this approach, as you can imagine, cannot really accommodate more complex events. In fact, it can only do linear sorts of inferences of trajectory, can't do any bifurcations or anything. So if we don't have a linear process, if we have a multifurcating or bifurcating process, how do we deal with that exactly, but still use kind of this simplicity and elegance of a principal curve to draw out trajectories? So we can adapt the principal curve to accommodate these more complex. In all different sorts of topologies, we can draw a single line connecting a given root node to a given terminal node. And essentially, what we could do is we could draw just a bunch of lines, going along each of them, and we can do some sort of comparison to see like which lines are the most likely to be occurring in the data set across, the given topologies, deconstructing more complex topologies into purely linear topologies. Now, the way, instead of testing like every possible connection, we can use the prior approach, the minimum spanning tree approach that connects clusters, and use that as a sort of prior to give us the space in which we want to experiment with drawing principal curves. And so in doing so, we can start to fit principal curves across all the lineages and start to figure out, all the potential different connections and start to suss out, what might be likely sorts of trajectories that we see. We can start to map it onto gene expression measures, for example, and start to look at those relationships and see what makes sense biologically and mathematically. Now, in all these cases, what the end result is going to be is that for each cell, we have a given trajectory unit. Now, that trajectory unit may be across any number of different lineages, different pathways within a given trajectory or whatnot, but we get these values, and what do we do with them exactly? Is we get into the huge variety of different downstream analyses that we can approach, but really what they all boil down to is drawing an association between this new trajectory time quote unquote axis and relating it to something else. So we're across a lot of different methods, we're essentially fitting a natural spline to the expression of each gene relative to this trajectory axis, and we can start to perform statistical tests that assess if there's some significant sort of trend, drawing on the wide body of work that has already done a lot of investigation in terms of these approaches. And we can start to visualize it using things like dot plots and heat maps. You've probably seen similar sorts of representations as the heat map on the bottom right. But suffice it to say, we're just looking for associations that make sense. And so this might be one way that you start to corroborate, if you have different results, as we saw in our previous analysis, that we connected cluster 1 to both to 2, 3, and 4 across different methodologies. We might start to look at these sorts of associations to see if there is some sort of more sensical, biologically interpretable sort of axis of variation that explains, might best associate with a given trajectory topology. Once again, I want to touch on RNA velocity. So RNA velocity is going to follow a lot of similar different considerations as we've drawn out here. And naturally, it starts from a different basis, using spliced and unspliced matrices, and does that in order to draw out the relationships between the cells. So here, for example, we can see that what I should say is that velocity analysis can be very complementary to what we just did. So in the previous approaches, you probably noticed that we don't have any arrows here. And that's because we cannot determine what the directionality is along these trajectories. We don't know if we're going to start from root node 4 or root node 7 or 8 in this representation, or even we might be starting from anywhere in the middle. We might be starting from 1 and going outwards. So that question can potentially be addressed by the usage of velocity RNA velocity. And what we can see is what sorts of states might be giving what the directionality of our results could be. So we can map these velocity results onto our previous analysis. So for example, you'll see on the right, t-SNE on the right-hand side, which is drawn based on the RNA velocity results, you'll see that there seems to be an intermediate population in the middle left part of the graph that seems to be going in both directions, seems to be going in both upwards along this tail and downwards along the other bottom-facing tail. So we can imagine that that might be indeed 2, and it gives rise to this sort of bifurcating process. RNA-seq, RNA velocity, in the absence of other a priori knowledge, could be very useful in helping us to define a route in our trajectory analysis. Now, so again, velocity analysis hinges on a lot of different assumptions, a lot more assumptions, and the key one is that transcriptional dynamics are similar across subpopulations, and naturally there's other caveats associated with specifically with the unspliced counts matrices, in that there's some observability and annotation sort of issues that may come up. So naturally, the gold standard is always going to be comparing to some experimental sort of setup and doing validation on that, or doing knockouts to assess branch points and whatnot, and their functionality and such. So yeah, so we have all these different approaches. We have these cool frameworks that can start to give us potentially new axes that explain a lot of the underlying processes and gene expression profiles, and really just highlights how powerful single-cell RNA-seq is, in that it's really a coagulation. It's an aggregation of a lot of different things coming together all at once that have enabled these really exciting new computational methods to come to the fore. So there's one other thing you walk away from besides that nice workflow that helps you pick a trajectory analysis method, it's that be careful where you apply trajectory analysis, because again, it's very easy to overinterpret. You're going to draw, if you have a pure noise data set and you apply a trajectory analysis method, you're going to draw a line of some sort. And so you need to be able to justify that line and be able to interpret it, make sense of it. So you can very easily draw a bajillion different trajectories, and only one is the one that makes sense. So yeah, thank you so much. Special thanks to, of course, HCA Latin America organizers for giving me the opportunity to present this sort of lecture and overview, and of course my mentor Rafael Guittardo, who it's been wonderful to work with. So yeah, thank you so much.
Thank you, Robert, for this excellent, very enlightening talk. We have some questions, so I would go directly to the questions, so we may proceed. We have a question from Elena, who says, this might be a truly basic question, but which transcripts are used to perform trajectory analysis? Is it only coding, poly-A transcripts? Could non-coding RNAs or other non-coding genomic elements that are transcribed be used?
Yeah, so are you specifically, so if you're referring to velocity analysis, RNA velocity is using spliced and unspliced, and it's drawing on, well, okay, let's back up. So in generating a counts matrix, it really depends, what features you get in that counts matrix is going to depend on your genome annotation. So your genome annotation could be only protein coding genes, or it could be an exhaustive compendium of everything under the sun that we know about. So these analyses are all predicated on performing feature selection prior to dimension reduction. And that's just because dimension reduction works best if you have a lot more information in your data set and aren't dirtying it with sort of like noise, as you might expect. I mean, a given, like a T cell, for example, really only expresses 2,500 genes or so, maybe even less, like reliably. And if we're including a large compendium of 30,000 different vectors, that noise from the other non-2,500 genes that a T cell expresses is going to dirty up the dimension reduction representation. And so feature selection is key. So the long story short is it can be whatever you want. If you think poly-A or ribosome genes are of interest and valuable, then give it a try. Nobody's stopping you.
Yeah, actually, also from Elena, we have a related question. Has anyone tried to put together velocity analysis with ribosome profiling, trying to figure out the continuity between the transcription and translation processes in relation to cell identities?
I'm not aware of any work that has covered that particular aspect. So, I mean, that definitely sounds like a promising avenue. And with regards to specific biological sort of setups or questions. So yeah, I mean, sounds feasible, sounds exciting.
Okay, thank you. We have also a question from Benjamin Pizarro. Is there any consortium to try to, perhaps it's consensus, to try to normalize and standardize these methods? So
yeah, so actually so yeah, you can imagine that each method has its own way of doing trajectory analysis, but like I have presented, there's kind of a general framework that that is shared by all of these methods, right? You know, drawing the curve, creating, you know, what are the calculating distances between communities and cells and stuff like that. So this review paper actually is also associated with the software suite called Dynverse. So D-Y-N-V-E-R-S-E. And if you Google that, you'll land on a splash page that has a bevy of trajectory analysis sorts of frameworks and tools that allow you to do things like benchmarking, run all these different trajectory analyses, all with a common API. So, you know, you can run 5 different sorts of methods with a single coding approach. So really, I highly recommend checking it out. Yeah.
okay, thank you. What can we do if the minimal spanning tree structure is not satisfied? So for instance, if there is loops, is there a way to disambiguate trajectories containing some loops?
Yeah, so yep, like you said, a minimum spanning tree is not going to elucidate loops that, you know, cycles that may be actually present in the data. So this is definitely a limitation of that algorithm. So like once again, in the review paper, I would highly suggest looking at methods that can accommodate cycles and may specifically focus on elucidating cycles, and you know, combine that with other methods that may decipher other topologies and combine the results from the two, similar to, you know, how we used complementary approaches, the principal curves approach and the minimum spanning tree approach to better dissect potential trajectories. We can use different software suites that are focused on different types of trajectories to create a unified sort of representation. Yeah. So I think that's
okay. Thank you. Benjamin Pizarro also asks, what other graph theory tools can be used for this kind of analysis? Shortest paths, node connections, centralities?
Yeah, any of those. So there is related work. There is other work. Just like I mentioned, you know, there's been a whole bevy historically of approaches to this. It still is an active area of research, although definitely very saturated. And one way that trajectories have been inferred is using actually graphs and force-directed graphs in particular. And some groups have observed that these force-directed graph layouts do align to, you know, trajectories and very naturally order the cells into like this trajectory sort of configuration without too much more work. And so definitely there's other, this is one example of a more graph-based approach to the problem. And certainly, you know, like I mentioned, with regards to clustering in particular, there's going to be many different ways to define the clusters. The 2 algorithms in particular, the Leiden algorithm and the Louvain algorithm, to identify communities. And there's a whole ton of others. You could use hierarchical clustering and whatnot. So it really, you know, once you understand this framework, this skeleton that you need to walk through for trajectory analysis, it really becomes possible to start making your own sort of ad hoc approach and switch in and out all these different sort of, you know.
Okay, well, thank you. Well, that's somehow a redundant question, but anyhow, if you want to add something, is there a method that could detect cycles?
Yeah, once again, I would recommend looking at the Dyneverse sort of paper by Salins et al. I'm looking at the table, and it seems like there's software called Angle that may be pretty performant. So I would probably start there if I was doing that sort of analysis.
Okay, thank you. And a final question. Is there a database of trajectories in web for specific processes like cell cycle or apoptosis?
Yeah, that's a really good question. So you would think so. One resource I would look at is the Trapnell lab. So Trapnell, guy who wrote Monocle, the first Monocle, and continues to maintain Monocle and develop it through his lab. He released this mouse atlas, and in that, using single cell or single nucleus RNA-seq, some sort of single cell transcriptomic approach, and it covers a lot of the different organ systems of mice. And within the results, his group performed trajectory analysis. So I would suggest looking at that as maybe as a starting point. I haven't used it myself. I've just heard Cole Trapnell talk about it. So I'm not sure how available or how easy it is to use, but those sorts of areas is something I would start to look at. Yeah.
Okay, well, thank you. We really enjoyed your talk, and thank you for joining us in this workshop, Rob. Let us move on to the next speaker. Thank you, Rob, again. And it's also my pleasure to introduce Professor Harikrishna Nakshtari. He's the Marian J. Morrison Chair of Breast Cancer Research and the Professor of Surgery, Biochemistry, and Molecular Biology at the Indiana University School of Medicine. He's also the Associate Director for Education at the Indiana University Simon Comprehensive Cancer Center, and he serves as the research careers scientist at the VA Rootbush Medical Center. Dr. Nakshtari studies the molecular drivers of therapy resistance in breast cancer. His laboratory was the first to identify the role of the protein complex NF-kappa B, which controls genes that respond to environmental stress and infection in triple-negative breast cancer. He has also identified markers that may predict response to anti-estrogen therapy. Utilizing normal breast tissues of women of different ethnic background, his group has discovered genetic ancestry-dependent heterogeneity in the normal breasts, which has important implications on how tumors are characterized for genomic abnormalities. And his recently published studies may enable to understand why hormone-responsive breast tumors are more common in women of European ancestry, and why triple-negative breast cancer are aggressive in women of African ancestry. Additionally, his group is mapping the normal breast as well as the breast of BRCA1 and 2 mutation carriers at the single cell resolution using single cell sequencing techniques. These efforts may lead to classification of breast cancer based on cell of origin of tumor, and he's using systems biology approaches to understand organ-specific breast cancer metastases and developing patient-derived tumor models that reflect organ-specific metastases and therapy resistance. So it's a real pleasure to have Professor Nakshtari with us. So we have your attention, Professor Nakshtari.
Thank you for the invitation. I hope you can hear me correctly. Can you hear me?
Yes, perfectly.
Okay, I will share the screen. Thanks again for the kind invitation and introduction. And what I'm going to tell you today is some of the work the lab is doing, and hopefully you can see the screen clearly. Right? Okay, good. So once again, thank you. And today I'm going to focus mostly on the work that we have done at a single cell level using the normal breast. The BRCA1 and 2 data are still being processed. It is not ready for prime time yet. The single cell data of the normal breast, which we just published in March. So the paper is already available. The data are also publicly available. I'm going to go and let you know where exactly it is available. So what I'm going to tell you today is the source of tissue for our study. This is the most critical element in my talk, because unlike the most other studies that you heard about from the normal, in our case, we are using really a true normal breast tissue that is donated to us by healthy women for only for research purpose. I'm going to explain to you how we are getting this tissue, then how we generated a single cell map of the normal breast. In fact, after hearing the previous talk, we are not too deep into bioinformatic analysis. Mine will be what the 10x genomics provides. I'm sure there is a scope for further analysis of our data that are already publicly available. How we figured out the reproducibility of our clustering. Then, since my basic interest is in breast cancer, what we then did was to derive cell of origin signatures based on the clustering we did with the normal breast. Then we applied to the breast cancer data sets to figure out, are there any correlation for the breast cancer subtypes and the cell of origin signatures we developed. And I'm going to briefly go over what we are doing now and what we plan to do next. So this is the outline of my presentation. So the source of the tissue is from a bank called Susan G. Komen Normal Tissue Bank at IU Simon Cancer Center, which we think is the only source where breast tissues from healthy women are collected only for the research purpose. We have distributed this tissue to over 17 countries for hundreds of labs, this tissue. What we do is we collect the samples. They are collected using a standard operating procedure. That's one of the biggest thing about the biorepository, because if you start collecting the samples from different places and start analyzing, you have the risk of using the samples collected with the different standard operating procedures. In our case, we have standardized the procedure. We collect the breast tissue from the upper left quadrant so that the location of the tissue is kind of same in most cases. And cold ischemia time, that is from the time the tissue is collected and then it goes back to ice and then frozen away, is 6 minutes. So that's a critical element when you think about the single cell analysis. We do examine all the tissues histologically to make sure that although women do not feel that they have a breast cancer, but there may be some abnormality lurking underneath. So we make sure that our tissues are devoid of that. We collect extensive data from these women, including their BMI, the number of pregnancies they had, their contraceptive use, alcohol consumption, tobacco consumption, any relatives with breast cancer, what is the relationship, and whether breastfeeding or not. There are multiple pages of information we collect from these women so that you can go back and relate whatever we observe into the demographic data. And then also based on the family history, and for several of them, we have the radiographic images of the normal breast. So we can classify the tissue that we collected are either from average risk women or high risk women based on the Trager-Kozik method. And we have DNA from all of these women and from their blood that has been used to map the genetic ancestry. So we are not only relying on the self-reported race information, but also we have mapped them all using the genetic ancestry of 41 new SNP, but we are in the process of extending this to a latest version where you can classify human population into 17 groups based on the genetic ancestry that the study came out just 2 weeks ago. We are kind of moving in that direction to reclassify our samples using this extensive genetic ancestry information. And there are few women who have donated the tissue twice, once now and after 2 to 5 years. So we can use those tissue for age-dependent changes in the breast, in the normal breast. So these are this is the advantage of the tissue bank that we have in our place. Just to briefly explain, this is the typical scenario. As soon as generally we have these events happening on Saturday, we have done this collection in multiple places, not just in Indianapolis where it is located. We have done it in Houston, we have done it in New York, Chicago, and California, we have done that. So the volunteers collect all the information that's entered here, and the basic blood drawing, everything is done with the patient. And these are the volunteers, they are processing the tissue. These are the types of cores that we get from women. And then two cores are cryopreserved. I'll come back to that in a minute. Then we'll have frozen section as well as paraffin embedded FFPs, the paraffin formalin fixed tissue as well. So we have several cores, and those are processed immediately and put into the storage. So this is not a few samples that we have. We have 5,641 frozen samples, 5,044 FFPE, and my lab developed a method where you can cryopreserve this tissue so that you can develop live cultures out of them after a few months, whenever you want it. So we have 1,280 of these samples. These are the samples that we are using for our Chan Zuckerberg funded single cell atlas in most cases. Because of the COVID-19, we can't do the new collection anymore. We are relying on these samples. They give exceptionally nice data. Then we have DNA and blood from around 11,000 women, from which we have serum has been collected from 6,000, close to 7,000, and plasma from 8,000. Again, all of these samples are collected using a same operating procedure. That means there is a uniformity in the quality of samples. So you don't have to go from lab to lab or institution to institution, then worry about are the qualities the same. So if you have a longitudinal study you want to do, or any of these studies, this is one of the very well annotated tissue bank. In fact, National Cancer Institute is using some of our materials for their own research. With respect to demographics of this one, 74% of them are from Caucasian women. The next biggest group is 17% African American, and Asian is 3%, and Native American Alaskan is 1%. The remaining, the others are unknown. 2% are mixed race. Again, the non-Hispanic represents 92%, Hispanic represents 8%. So these are based on the percentage of minority tissue samples. Again, Asian is 3%, slightly above national average, although we feel we need more because Asian is not one. There is East Asian, South Asian, Southeast Asian. They have a different genomic diversity. So we need to collect more of the samples. And again, we have done pretty good in collecting the sample from African American or Black. Hispanic also 8.7%, but again, Hispanic itself is not one genetic ancestry group. There are multiple groups. We still need to work on diversifying these samples. So what have we done with these samples? These are the sample distribution pattern, the frozen tissue, tissue sections, serum, plasma, DNA, and cryopreserved sample. Over the year, the demand for the tissue sections has been going up. For example, in 2018, we sent out to researchers 3,960 tissue samples. My lab has created a tissue microarray out of this, where we have made tissue microarray, although they are not as good as the tumor microarray because these are tiny pieces of tissue. After multiple cuts, they fall off. Although what we have done is to generate tissue microarray combining of the healthy women breast tissues of Caucasian women, African American women, and Hispanic women, and combine that with the tumor and tumor adjacent normal from our another bank. So overall, about 300 samples in a TMA that one can analyze. What is the difference between a normal tissue versus tumor adjacent normal, and a tumor adjacent normal versus tumor? So it gets you a broader comparison, because our studies and many other studies have shown tumor adjacent normal is not a normal tissue. So real normal has to come from healthy women, and this type of TMA allows you for this comparison. So, and we follow up these donors as well. We have connected these samples to the Indiana Death and Cancer Registry to update records and discover participants who may have subsequently developed breast cancer. And we have every birthday, we send out a card asking them, are there any changes in the health history? So just let us know so that we have, we get response from 50% of them. So we keep on adding the health data. And unfortunately, out of, which is again almost as good as the normal regular population, out of 5,000 women who donated, 120 of them have developed cancer after donating. The donation has been done. So there is an extensive amount of study that we are doing with these samples, which were we have what we call pre-cancerous sample. Before they develop the cancer, 2 years, 3 years, or 5 years before, we have those samples. We are comparing them with those who did not develop breast cancer. So it's a wide variety of tissues that we have. It's an ample resource for many of you who are interested in studying this. As of December 2019, we have 120 germline mutation carriers who have donated this tissue. And this cryopreserved tissue has been very helpful for us. And what we have been able to do is we have to able to generate the cells and generate cell lines, immortalize them. As many of you know, if you're working in a breast cancer field, everyone works with one so-called normal cell line called MCF10A. But what we have been able to do is generate multiple cell lines from the healthy breast tissue. These are all genetic ancestry mapped. For example, the top of the 4 cell lines are all from European ancestry women. They carry more of European ancestry DNA. These are from African American ancestry SNPs. These are for Hispanic women. Again, this is not homogeneous population, as I mentioned before. There are multiple populations, multiple distribution pattern with this, the 41 nucleotide, single nucleotide polymorphism. These are some of the publications coming out of these cell lines. These cell lines have been distributed to more than 20 or 30 labs so far. There's a heavy demand for that. We are ready to give it out to anybody. So this is how we created this normal breast tissue resource. Having created this tissue resource, our next step was to use them for single cell genomics. You may ask the question, why did you go this far? Why can't we do it with the reduction mammoplasty samples? Why can't we do it with the samples that are from the discarded after the surgery, tumor adjacent normal? There are multiple reasons why we felt that the breast biopsies from clinically healthy donors are absolutely essential. For example, as I said, reduction mammoplasty samples are the routine tissue. If you compare the reduction mammoplasty samples with that of this, what is that, no histologic abnormality, there is a non-proliferative disease, atypical hyperplasia. On the top is the donors to our bank. 65% of them have no histologic abnormality. If we compare that, only 12% of reduction mammoplasty samples had no histologic abnormality. 71% of them had non-proliferative disease compared to 31% from our case. So there is an enormous difference in the quality of the samples that we get with our samples. Breast biopsies are collected from healthy donors of diverse genetic background. So that's an important consideration. And we collect the tissue from the same region, so that way the heterogeneity is less. Tissue collection to single cell RNA sequencing within 3 hours. Many of you know the time lapse between the tissue collection and single cell RNA sequencing, as well as the digestion methods, affect the quality of single cell RNA sequencing data. And it allows the cryopreservation, rapid cryopreservation for future analysis that we have been doing now. But there are caveats. Limited tissue to work with. Epithelial cellularity varies with significant number of samples being enriched for adipocytes. So that is one of the caveats you need to take into consideration. And out of 30 samples we sequenced, maybe 15 of them came out okay and publishable data. So there is a lot of loss, and the number of cells you sequence per sample is not as much as if you have a whole big breast tissue that comes from reduction mammoplasty, where you can chop into multiple pieces and then do collect a lot of cells by flow cytometry and then do the analysis. In our case, it is not. We are doing the limited tissue. We want to limit the time lapse between the tissue collection and the first cDNA synthesis so that most of with the hope that RNA truly represent what is in the cell at the time of tissue collection, rather than impacted by the harsh treatment condition. Here is the data from 5 samples that I'm giving you. So these are the demographic information. And this was done with the freshly collected biopsies. We had the samples collected in the morning of a Saturday. My lab itself purchased a single cell sequencing machine costing about $65,000. As soon as the samples came out, we brought into the lab, analyzed, and we collect all of this data, including Tyrer-Kauzik lifetime risk score we developed. A score of about 25 is considered, about 20 is considered poor, and there are 2 women in this collection are above that risk. You can see there is a family history of risk. We have the BMI information, how many times they were pregnant before the tissue donation, and this is their aged collection. What you see is a single cell map, and you find 3 different epithelial cell type clusters, 3 fibroblast-like clusters, and 2 NK cells, and these are the epithelial cell clusters. These are endothelial cells, these are epithelial cell clusters that are available. 13 epithelial cell types, 3 endothelial cell types, and fibroblast-like cell types. What are these cell types represent, particularly for the presentation, and at the moment our focus is on epithelial cells. I don't want to confuse the people by analyzing everything at one single study. What we did was, went back to the literature. As some of you in the breast cancer field know, that in human breast, most of the studies of the human breast epithelial characterization involved flow cytometry-based sorting. What is typically used is the lineage negative population of the human breast, then use the EpCAM and CD49F. If the cells are only CD49F positive, they are considered stem or basal cells. If they are only EpCAM positive, then they are considered mature luminal cells. Those who are double positive are considered luminal progenitor cells. Negative are considered stromal cells. In mouse, it's a different set of the primers. What others have also done was to use these parameters and then look into the basal cells, again, EpCAM low and the CD49F. What is the transcription regulatory network that is active in that? Luminal progenitor cells, there is a ELF5, EHF transcriptional activity, whereas luminal mature cells, CD49 high, CD49 low, EpCAM high cells, they have FOXA1 ESR network. What we did was we applied the cell surface markers and the signaling network to subclassify our epithelial cells into 3 basal or stem clusters that is represented here, 7, 5, and 9 clusters, and 7 luminal progenitor clusters, and that is represented here, the luminal progenitor clusters, and 3 luminal mature cell clusters. Again, here are this. Based on what you saw before in this analysis, these are clubbed into only one group, mature luminal 1, luminal progenitor 1, where the stem cells or basal cells as 1. Whereas with this analysis, now we know that there are multiple groups within the luminal progenitors, multiple groups within the mature luminal, multiple within the basal stem cell. How do we verify and make sure that what we subclassified makes sense with the existing literature? Let me give you an example of some of the markers that we looked into. And here is the marker CD36. It's predominantly the red color represents they are high in expression. This particular CD36 is expressed predominantly in the basal cell cluster. By contrast, SFRP1 is expressed only in the luminal progenitor cluster. You see few cells being positive in the mature as well as basal. By contrast, XBP1 is expressed at a high level at a mature luminal cluster. So apart from the markers that I showed you, cell surface markers, CD40 and EPCAM, the genes that are expressed within these clusters can further classify the cells into basal, luminal progenitor, and mature cells. The additional markers for the basal cells include, as I told you, CD36. You can see this NDUFA4 is subset of the basal cells expressed, but nothing in the mature luminal or luminal progenitor cells. CLDN5, that is claudin, one of the claudin, again expressed in the basal or basal stem cells. MECOM1, few cells within the basal cells. So you can further refine the cells, further identify the cells that are, the genes that are uniquely expressed in the basal cells of the breast. Moving along with the luminal progenitor, what came as a surprise is the keratin 14. For many, many years, people thought that the keratin 14 is expressed only in the basal cells. But in our hand, it's only a subset of the luminal progenitor cells that express keratin 14. I will come back to this in a minute when I go to the other set of studies. Keratin 17, it is again in a similar pattern, which has kind of overlaps with what we find with the keratin 14. Now, with respect to mature luminal cells, you have the XBP1, as I already told you. And these cells are negative for SFRP1. Previously described ANKRD30A is expressed mostly in the luminal progenitor cells. MUC1 is expressed in only one subset of the luminal mature cells, as well as PIP. So there are not all the luminal mature cells express this PIP4, but it's only a subgroup. What it tells you is that the luminal mature cells, as defined by EPCAM positive, CD49F negative, itself is a heterogeneous population, and that there are multiple cell types with the varying level of gene expression. So one can use XBP1 and SFRP1 and CD36 then to probably map the level of mature luminal progenitor, and stem cell in a section of the breast without having to go through all this flow cytometry or single cell. That's what we are attempting to do now. Now, one of the critical things that we all experience in the breast cancer field is the estrogen receptor. Estrogen receptor is a nuclear receptor. It's important for the breast cancer. It's a critical signaling molecule for normal breast development. Unlike in breast cancer, it is expressed predominantly in non-proliferating cells. By paracrine signaling, it controls the proliferation of ER negative cells. Although in the normal breast, it represents non-proliferating cells. In case of breast cancer, however, 70% of them express estrogen receptor. They are proliferating in response to estrogen treatment. That's the reason why anti-estrogen treatments are the effective treatment for breast cancer. But the problem has been, it has been extremely difficult to identify genes co-expressed the ER in the normal breast. As soon as you culture the breast epithelial cells, they lose their estrogen receptor expression. So in the normal tissue, we had extreme difficulty in figuring out what is co-expressed with estrogen receptor in the normal breast. And at least in breast cancer, pioneer factors such as FOXA1, GATA3 regulate genome-wide binding of ER. But we don't know it happens the same here or whether these pioneer factors are expressed along with the ER in the normal breast. So that's the reason why we gave a lot of attention to the estrogen receptor. And here is the one case, I'm showing a representative case where we were able to see the estrogen receptor in one cluster. This is the cluster that expresses estrogen receptor, all of them. And again, in this cluster, also some of the cells express estrogen receptor. But if you look into the GATA3, which is a pioneer factor for estrogen receptor, it is expressed at a low level in this ER positive, but there is a cluster without ER alpha that expresses this. What we found is there is TBX3 and PDK4, the previously uncharacterized proteins, are co-expressed with the estrogen receptor in this cluster. You can see the TBX3 is almost equivalent to that of the estrogen receptor as well as PDK4. I will come back to this point at the end, but what this study has enabled us is to identify at least 2 subtype of estrogen receptor positive cells in the normal breast, one expressing both the TBX3 and PDK4 along with the estrogen receptor, the other having lower level of these factors. So since we had the practical difficulty in collecting the tissue and doing the single analysis at the same time, because all you have to coordinate the tissue collections are usually on Saturdays, and need to have the lab personnel ready that day. It's not that easy. It takes about 4 to 5 hours once you get the tissue. So it becomes incredibly tough to manage with the people in the lab. So, and again, you cannot do one sample at a time. At least 4 samples has to be in one cartridge to make it reasonably cost effective. So considering that difficulty, we developed a cryopreservation method to store the tissue in for future single cell sequencing. If anybody is interested in how to cryopreserve, the protocol is available through Protocol IO. If not, you can send me an email out. I will send it to you. I have distributed this method to multiple groups already. So with that, what we did was we used new set of pooled 5 cryopreserved sample and a fresh sample. Nowadays, the lab has been doing with the pooled samples, 5 cryopreserved sample in one tube, so that your cost will come down. And here we are not interested in looking at it in an individual per se, although the current genomic technique will help you to even tease it out, even if you do a combined sample analysis. So we then integrated the analysis of all 11 samples of 2 sets and did an individual as well as integrated. And then we overlapped the gene expression between 2 sets of analysis to make sure that they represent correctly. Now we have increased number of cells, increased level of gene expression. So what happened in this case is that the number of clusters that we identified increased substantially. And this is again the basal stem cluster. In the previous one, you had only 3 clusters. Now we have about the 8 basal and myoepithelial clusters. Luminal progenitor clusters went up to 8, and there are 4 mature luminal clusters. So, and this is again the pooled sample set and the first set, both of them show a similar pattern of distribution. So with the increased number of samples, we were able to further classify them. We made sure that the expression pattern, as I told you before, CD49F expression is highest in the basal and basal stem cells, whereas they're intermediate in these luminal progenitor cells, pretty much absent in the mature cells. By contrast, the basal cells do not express EpCAM. Both the luminal progenitors and mature cells express EpCAM, which is pretty evident here. Then we looked into these EHF and CD117, which is again luminal progenitor. You can see the distribution pattern. The luminal progenitor cells expressing the highest level, particularly this one particular cluster seems to have very high level of EHF1. EHF1 is also involved in the alveolar differentiation. Potentially the alveolar cells may be coming out of this particular cluster. We don't know yet. And CD117, again, a luminal progenitor marker expressed mostly in the luminal progenitor cells based on our classification. And if you look into the mature luminal cells, this is the expression pattern of estrogen receptor. Not all luminal progenitor cells express. There is a low level of expression in the luminal progenitor cells, high in the luminal mature cells. Maybe I must have told you wrongly before. It is luminal mature cells here. There is an expression, but not all the cell type. The progesterone receptor, PGR, is downstream of estrogen receptor. It is induced by the estrogen receptor. Its expression is restricted to mature luminal cells, and it overlaps wherever it's expressed with the estrogen receptor. The GATA3, which is a pioneer factor for estrogen receptor, expressed very high in the mature luminal cells, but at low level in the luminal progenitor cells. FOXA1, by contrast, which is again another co-activator or pioneer factor for estrogen receptor, expressed predominantly in the mature luminal cells. GREB1 is an estrogen-inducible gene, also regulates estrogen receptor activity, is expressed only in a small subset of cells. TNSF, we have 11, which is a rank ligand expressed in mature cells, which is known to interact with, provide the ligand for the rank receptor in the luminal progenitor cell proliferation. Again, its expression is restricted to only a subset of mature cells. So what you need to remember is that not all mature luminal cells are estrogen receptor positive. That means only a subset of cells in the mature luminal cells are the source for the estrogen receptor positive breast cancer. With respect to keratins, again, keratin 18, 19, this 18 and 19 are supposed to be expressed in the mature luminal cells, to some extent in luminal progenitor cells, but not in the basal cells. Our study confirmed that. Keratin 18, the same pathway. What is known about KRT5 and KRT6B were thought to be restricted to the stem cells, but what we find them is their expression in a subset of luminal progenitor cells, but not in the basal cells. So this needs some more investigation in future. Keratin 17 is the same pattern. Keratin 14, as I told you before, it is expressed predominantly in the subset of luminal progenitor cells, unlike what was believed previously as a basal cell marker. ALDH1A3, which is considered as a cancer stem cells or metastasis initiating cell marker, only a subset of luminal progenitor cells express this marker. So we have been able to kind of pinpoint the expression patterns of various markers that have been studied only in the context of breast cancer before into individual cell type in the normal breast. So that is the progress we were able to do with this single cell analysis. Now, as I pointed out, my interest is in breast cancer. How can I apply the knowledge that I gained from this single cell analysis of healthy breast to predict the cell of origin of breast cancer? The next question is why knowing cell of origin is important. So these are the questions the lab has started to ask. The reason that is knowing cell of origin is important is because gene expression in cancer is influenced by cell of origin of tumors, adaptive signaling by cancer cells, and genomic aberrations. It is not one thing that determines the gene expression. It's the combination of all three that will tell you what is the gene expression pattern in cancer. And then also this study came out. What it claimed was that the cell of origin patterns, rather than the genomic aberration, dominate the molecular classification of cancer. So having this literature in mind, we thought, let us use this signature that we created representing these 23 epithelial cell types of the normal breast. So we created the gene signature corresponding to 23 clusters, applied these signatures to two major breast cancer gene expression data sets, TCGA and Metabreak, where we had the access to the gene expression in approximately 3,000 breast cancer samples. Then what we did was to determine the frequency at which signatures corresponding to each of these clusters are represented in these tumors, and then determine the impact on survival. This is a classic example. In all of these clusters, we had the genes that are uniquely expressed in these clusters are represented in the tumor samples, others were not. This is in the TCGA, this is in the Metabreak. And you can see the progression-free survival overall survival in each of these cases based on the tumor had a gene expression corresponding to one cluster over the other. What came as a surprise to us is that the majority of the breast cancers in case of this TCGA were classified in the cluster number 12, cluster number 17, and cluster number 19. Cluster number 12 and 17 are within the mature luminal clusters, whereas 19 is located in the luminal progenitor cluster, none of them from the basal cluster. With the meta break, a similar sort of data. Cluster number 8, which is also a mature luminal cluster, 747 cases, followed by cluster number 12, as in this case, and then is the cluster number 19. So the cluster number 19, which is a very tiny fraction within the luminal progenitor cells, are heavily represented in this particular breast cancer data sets. So I'm sure if you are a clinician, you know how we classify the breast cancer at this moment. And the major classification that is being done in the breast cancer field is using the intrinsic subtype or PAM50 subtype cluster classification. Basal-like breast cancer, Claudin low breast cancer, HER2-enriched breast cancer, luminal B breast cancer, and luminal A. Both of these are estrogen receptor positive with variable level of estrogen receptor expression and different kind of mutation. In this case, 40% PI3 kinase mutation, whereas here they express GATA3, FOXA1, and XBP. At a surrogate intrinsic and the clinical level, you class them as the triple negative, HER2 enriched, luminal B like HER2 plus, luminal B like HER2 minus, luminal A like. Of course, the proliferation rates are different. Triple negative is higher. They are high grade. They express basal-like gene, whereas this luminal A express ER and ER related gene and low grade. What was proposed in 2009 was that in the normal breast, these are the types of cells that are present: stem cells, bipotent progenitors, luminal progenitor, late luminal progenitor, and differentiated cells. What was proposed that the Claudin low breast cancers may be coming from the stem cells, basal-like may be coming from the bipotent progenitor and luminal progenitor, HER2 coming from the late luminal progenitor, and the differentiated cells are the source of origin for luminal B and luminal A. If this model is correct, the luminal A breast cancer in TCGA or metabreak should come from, all of them should be coming from the differentiated luminal cells. But what we found out by contrast is that the luminal A breast cancers, there are 280 of them have the gene expression similar to a mature luminal cluster 8. 159 has the mature luminal cluster 12, but 184 of them have the gene expression similar to a luminal progenitor cells. So that means all luminal A breast cancers are not originating from the differentiated luminal cells. Some are coming from the luminal progenitor cells. And those coming from the luminal progenitor cells have a better outcome than those coming from the mature luminal cells. And the same thing with the luminal B breast cancer. I think okay, now it's coming back. Same with the breast cancer. By contrast, even the basal type breast cancer, which was originally thought to be coming from luminal progenitor, is coming from all three clusters: N8 mature luminal, N12 mature luminal, N19 a luminal progenitor. Again, those coming from the luminal progenitors have better outcome than those coming from the mature lumen cells. By contrast, the HER2 positive breast cancers, if they are originating from the luminal progenitor cells, they have worst outcome compared to those which are originating from the mature luminal cells. So depending on the type of cancer, you have a different outcome based on their cell of origin. HER2 positive, if it is from a luminal progenitor, they have a poor outcome. So this is with the data sets. Just recall what I told you before is with respect to estrogen receptor, that's one of the lab focus. Estrogen receptor, we have it co-expresses TBX3 and PDK4 in one of these clusters. here, right here, this cluster expresses all of them, whereas this cluster, there is estrogen receptor, low level of PDK4, and low level of TBX3. So what we then did was we have a tissue microarray of 600 tumors, and we have 15 years follow-up on these patients. Then what we did was we did the immunohistochemistry for estrogen receptor, TBX3, and PDK4. Did the survival analysis. PDK4, if it is low expression, they are associated with the worst outcome. When the tumors have estrogen receptor, TBX3 high, PDK4 high, they had better outcome than if the tumor had ER positive, estrogen receptor positive, TBX3 low, PDK4 low. They have worst outcome. What we think is there are two types of cell types within the breast which express estrogen receptor, from which the estrogen receptor positive breast cancer may originate. If those which are originating from this cluster with all three of them, estrogen receptor, TBX3, and PDK4, that is associated with a better outcome. These data have been published. This just came out last month in Cell Report Medicine. And again, this is the cartoon version of it. The majority of the breast cancers originate from these three clusters, cluster 8, 12, and 17 of the mature luminal cluster. This tiny cluster within the luminal progenitor cells, the 19, is the other source for the breast cancer origin. And this, all the data have been already given to the Cellex gene maintained at Stanford. You guys can go into this site and then play around with the gene of your interest, figure out where exactly it is expressed in the normal breast. So this is an open resource that is available to all of you. The next question we have been working on is linking expression pattern of genes associated with the breast cancer risk alleles, and multi-omic analysis where we are combining HCA with RNA-seq as I speak. This morning only we have a set of samples which are already going through sequencing where we are combining the HCA with RNA-seq. This analysis will be expanded to include samples from Ashkenazi Jewish, Native American, and Asian women. We'll be doing approximately about 40 samples corresponding to these particular groups. Or the long-term goal is to come up with the spatial proteomics with antibody-based multiplexed assay so that we can take a section of the tissue from the healthy breast and analyze and figure out how many cell types exist. This is just an example of where we are trying to link into the breast cancer risk, the risk associated gene, how exactly they are, particularly the EQTL, decreased expression, decreased risk. This expression of these three genes are everywhere. It is not unique to a particular cluster. By contrast, the ER negative breast cancer, increased expression of these genes are associated with decreased risk. In this case, only the estrogen receptor is the only one which is uniquely expressed in a particular cluster. And the ER negative breast cancer, decreased expression, decreased risk. We have this CCDC 170. It is expressed only in this cluster. In fact, then this gene is right next to the estrogen receptor. They have identified in 7% of breast cancer, there is a fusion of CCDC 170 to estrogen receptor located next to the estrogen receptor one. So what you can do is if you have an expression QTL, which is, then if it is associated with breast cancer risk, you can go back and see whether this gene is expressed in the cell type from which the breast cancer originate. That's why having a mutation or SNP which alters its expression is associated with the risk. So you can go back and do that. The KCNN4 is mostly in the luminal progenitor cells. With respect to the spatial proteomics, we have started using single cell proteomics, 6 markers from a single slide with the normal breast tissue. With the Kodaks, we just did a trial run with the antibodies for CD45, CD2, CD19, CD38, CD104, CD11C, and then the DAPI staining. You can see the very clear staining of the basal cells with the CD104 markers here. So we are extending the studies further. So that's all I have for you, tell you today. Again, this is not a single person work. There are multiple people involved in this project. This is our team from different groups. And of course, my sincere thanks to countless number of women who donated breast tissue, as well as volunteers who facilitate this tissue collection. Usually in our tissue collection, we collect about 120 samples from 120 women. That needs around 200 volunteers, including from the surgeons who take out the samples to somebody who sits in the front desk and registers them. So it's a massive operation for us to do a tissue collection. And again, there are countless number of women through word of mouth, they spread the information to others, not force them, encourage them to donate for the breast cancer research. Again, those who donate may not benefit from it. These are all doing it for their future generation. And these are the funding sources that are helping various projects in my lab. This particular work is funded by Chan Zuckerberg as well as Catherine Peachy Foundation of the Heroes Foundation. With that, I stop and take your questions.
Thank you, Professor Nakhtari. Extraordinary talk. We are really grateful for you to have shared with us such a comprehensive view of both the basic and the clinical aspects, as well as the single cell issues of this work, and an impressive amount of work. So thank you for sharing this with us. We have a number of questions, so let me go straight on through them. Marco Antonio Fonseca asks, most of breast cancer classifications are focused on immunohistochemistry, microarrays, and bulk RNA-seq data. What are your perspectives about new breast cancer classifications focused on single-cell RNA-seq data analysis?
Yes, I think, again, if the single-cell RNA-seq will tell us most likely the cell of origin of tumors. So that cell of origin-based classification is something I hope will come in future. Probably that will help us to figure out, based on the cell of origin, what are the propensity for metastasis. Right? In a breast cancer, I don't worry too much about the primary breast tumor. We all worry about the metastasis, right? My hope is that the single cell analysis based studies, both at the genomic and RNA level, will help us to figure out whose tumor may have already metastasized, which is not detectable with the current technique. That's what I'm hoping this research will lead to.
Thank you. Regarding the several population groups, it says, wonderful multi-ethnic approach. Have you been able to find out whether ancestry somehow correlates with cell type repertoires or with cell type abundances?
Yes, this is not presented in this work. We have shown previously that it came out in Clinical Cancer Research in 2019, that African-American women have a unique type of cells which surrounds the duct. They are ZEB1 positive cells. They are relatively higher in the normal breast of African-American women compared to women of European ancestry. The number of these cells increase in the breast of women of European ancestry only if they have cancer. That is one thing we have found out. And then within the African-American women, that's another DoD funded study we are doing. If they are of sub-Saharan Africa, they carry a mutation in a gene called DUPI or ACKR that protects them from malarial infection. But the same genetic aberration, or not aberration, I would say, it's not an aberration, inherited change in the genome. If they get breast cancer, they have a worse outcome. And we have determined that the normal breast epithelial cells in a DUPI carrier have a different biology than those who are non-DUPI carrier within the African-American population. Yes, this genetic ancestry level mapping is really helpful in distinguishing the normal breast biology.
Okay, thank you. We have a couple of questions also related with ancestry. Thank you very much, says Camilo Villaman. Since ancestry is related to predisposition to certain types of diseases and cancer, do you think that there is any possibility of determining ancestry at the single cell level? Also, how does cell mosaicism affects the results?
I mean, the ancestry, you don't have to use the single cell level at all. You can take the blood DNA, and there are defined markers to determine the ancestry. As I pointed out in last week, the Cell paper that came out, where they were able to group the population in New York City into 17 ancestry groups. So there is an easy way how we can do that without doing the single cell to map the genetic ancestry. Yeah.
Okay, also related and perhaps already answered, it says, does the ethnic group was determined by geographic distribution or by ancestry genetic characterization, like mitochondrial markers or Y chromosome markers?
It is done using the 41 single nucleotide polymorphism ancestry marker panel. This is a older panel that is a 108 SNP panel that a lot of people are using. Now we are moving to this 17 ancestry markers, which requires the global ancestry. There's another term into that, so the global one, that's what we want to use moving forward. It's a lot more computational strength. And I just got an email that you need to increase the capacity, the storage capacity to analyze the data. So I need to figure that out. So we are further expanding and refining the ancestry marker, particularly the Hispanic population, as I showed you in one of the figure. And I get criticized whenever I use the term Hispanic and genetic ancestry right next to it in my grant application, because it is not one ancestry, right? So redefining that is very much critical if you want to base your cancer characterization, treatment decision based on the genetic ancestry.
Okay, and related to this, do this panel of ancestry informative markers you're using accounts for, let's say, mixed populations? Because in Spanish people, in Hispanic people, that happens.
Yes, exactly. Right. Yeah. I mean, that's what is very, very critical. I didn't tell you, I didn't include those data within the Latina population, which you identify with the single nucleotide polymorphism. If there is a lot more ancestry from Native American, there is a protective allele against breast cancer, right? That gets missed out if we just club them all together. There may be a group of women within the Latina ancestry, they are protected against breast cancer. By studying the breast tissues from those women, we may learn a lot more about how to protect against breast cancer. So that's why the refined genetic ancestry mapping is very, very critical as we move forward.
Okay, thank you. Thank you for sharing so much about your experiences with biobanks and the feasibility of using frozen samples. Do you look at the outcome of patients based on the clusters you find? Do you look for patients in the biobank with similar characteristics for validation?
So this biobank is mostly for the healthy breast tissue. And the tumor data that I presented to you is from a different biobank, which only collects the tumor tissue. So these are not connected in one way or the other. So it becomes a bit hard. I think once we keep refining these signatures, I think we will be able to apply it to the tumor samples when we get in what we get in the clinic. Yeah.
Okay, thank you. And also related somehow with biobanks, Fabio Takeosaro asks, well, he says, great presentation. Does the cryopreservation method to store tissues for future single-cell RNA studies preserves the cytoplasmic RNA?
Yes.
Is there any con to use this methodology as compared with fresh tissue? How much time is possible to use this after store? It says. So
exactly. I showed you one of the piece of data. The first set is from the fresh tissue. The second set is from the cryopreserved. They map exactly on each other, top of each other. There's no difference at all. And this was done with the cytoplasmic. Okay. In the sense, the total cell, we only took the live cells from the cryopreserved samples and doing it. By contrast, today what we are doing, where we are doing the multi-omic analysis, where we are doing ATAC-seq and RNA-seq from the same sample, we have moved to the nuclei. That is also from cryopreserved samples. So the cryopreserved samples will allow you to do simple RNA-seq as well as multi-omic analysis.
Okay, wonderful, wonderful work, wonderful talk. Thank you very much, Professor
Thanks for the
Nakti.
opportunity. Yeah.
Yeah, thank you. And well, with this, we end our afternoon session for today. I don't know whether we have some announcements for today or we are just moving on to this afternoon session. Patricia?
Yes, I think we could give some announcements for those who are here. Let me see if I can share. Yeah. So yeah, well, would like to thank everyone who joined us for this third day. Thank our speakers, and I'd like to remind you that there will be recordings made available of all the talks, and they will be at the YouTube and Bilibili sites of the HCA. So all the talks will be available there for those who cannot really join the afternoon sessions. We have